In the quiet beginnings of life, before there are tissues, organs, or even the faint outline of a body, there exists a cell unlike any other. This cell does not yet have a fixed identity. It is not committed to being part of the heart or the skin, nor has it chosen to become a nerve or a muscle. Instead, it rests in a state of openness, carrying within itself the possibility of becoming many things. This cell, and the many that follow in its line, is what we call a stem cell. To drift into the study of stem cells is to wander into one of the gentlest mysteries of biology. They are not loud in their function, nor dramatic in their presence. Rather, they are quiet foundations. They remain in the background, patient and adaptable, waiting for signals that tell them what is needed. Some may wait for years, resting in bone marrow or skin, until the moment comes when the body calls upon them to act. When they do, they rise to their purpose with a calm precision, becoming whatever the body requires. It is soothing to imagine these cells as small seeds scattered throughout the body. Some lie dormant in quiet corners, like seeds in the soil waiting for rain. Others are more active, continually dividing, replenishing the tissues that wear down with time. They are never rushed, yet they are always ready. Their presence assures us that the body carries within itself the means of renewal, the capacity for healing, and the quiet resilience of life. Every human being begins as a single cell, formed in the union of egg and sperm. From that one beginning, the miracle of division unfolds. One cell becomes two, two become four, and soon there is a cluster, a community of cells working in harmony. Some of these cells quickly begin to specialize, forming the earliest outlines of body parts, but others remain unspecialized, holding back, keeping their openness. They are the first stem cells, pluripotent and filled with potential. They can become any part of the body, from the beating muscle of the heart to the delicate tissues of the eye. These early stem cells are remarkable, yet their story does not end at birth. As the body develops and matures, many stem cells commit to specific roles, but not all of them do. A population remains, scattered through tissues, where they continue to serve quietly throughout life. These are the adult stem cells, less versatile than their embryonic counterparts, yet still invaluable. They ensure that blood is replenished, that skin renews, that wounds heal, and that the body retains its capacity for repair. One of the most calming ways to picture stem cells is as a kind of living reserve. Imagine a library filled with blank books, each one waiting to be written. Most of the volumes on the shelves already hold stories, tales of muscle, skin, nerve, or blood, but the blank books remain, unmarked, ready to become any story the body requires. Stem cells are those unwritten pages. When a tissue is damaged, or when a function must be restored, the body reaches for one of these blank volumes, writes upon its pages, and creates a new cell perfectly suited to the task at hand. The power of stem cells lies not only in their ability to differentiate, but also in their ability to renew themselves. While most cells of the body have a limited lifespan, stem cells can divide again and again, producing both copies of themselves and specialized cells. In this way, they maintain their own numbers while also contributing to the tissues that need them. It is as though they are both the guardians of potential and the workers of repair, balancing preservation with action. The story of stem cells is also the story of discovery. For most of human history, their existence was unknown. The body healed, blood was replenished, skin renewed, but the hidden actors remained invisible. Only with the development of modern science did these quiet players step into view. Researchers peered into bone marrow and found cells capable of rebuilding blood. They observed the embryo and recognized cells that could give rise to the entire body. These discoveries opened the door to a new era in biology and medicine, one where the body's own tools of healing might be understood and guided. And yet, Beyond science, there is something deeply calming about the very idea of stem cells. They represent not only the mechanics of biology, but also the essence of renewal. They remind us that even within wear and change, there is a possibility of restoration. A cut on the skin, a broken bone, even the daily turnover of blood, all are moments when stem cells step quietly forward to ensure continuity. They embody resilience, patience, and the gentle persistence of life. As you rest with these thoughts, you can imagine stem cells moving unseen through the rhythms of your own body. They are in your blood, ensuring its flow continues. They are in your skin, making sure the surface renews itself. They are in your marrow, waiting patiently, ready to step in whenever they are needed. They are small, but their role is vast. They are silent, but their influence is constant. In the end, stem cells are not simply cells. They are reminders of the body's quiet wisdom. They show us that life is never entirely fixed, that change and renewal are always possible, even in the smallest of forms. They are the blank pages carried within us, the seeds of healing, the companions of every moment. To learn about them is to learn about the capacity for renewal that resides in all living things, a truth as gentle as it is profound. Part 2. The building blocks of life. When considering stem cells, it helps to step back and remember that all living things, no matter how large or small, are built from the same foundation. That foundation is the cell. Cells are the building blocks of life, tiny compartments that carry out the processes needed for survival. In humans, there are trillions of them, each with its own role, its own task, its own quiet contribution to the whole. And yet, before a cell becomes specialized, before it knows whether it will become part of muscle or skin or blood, it begins as something more open and more flexible. It begins as a stem cell. A stem cell, in its earliest form, is like an unopened possibility. Imagine a seed that has not yet sprouted. It does not yet show leaves or roots, but it contains within itself the potential to become an entire plant. In much the same way, a stem cell contains the potential to become many kinds of cells, depending on the signals it receives and the environment it finds itself in. This potential is what makes stem cells so essential to life. They are not locked into one role from the start. Instead, they wait, listen, and they respond when the time comes. The body is built from many types of cells, each highly specialized. Muscle cells contract to allow movement, whether that movement is the beating of the heart or the bending of an arm. Nerve cells transmit signals, carrying messages from the brain to the rest of the body and back again. Blood cells carry oxygen, fight infection, and help with clotting when injuries occur. Skin cells form protective layers, shielding the body from the environment. Each of these cells is unique, yet all of them can trace their origins back to stem cells. Without stem cells, the diversity of cell types that sustain life could never exist. At the heart of this transformation from potential to purpose is the process of differentiation. Differentiation is the gradual narrowing of possibility, the moment when a cell begins to take on a particular identity. A stem cell, at first open to all possibilities, begins to receive signals, some chemical, some physical, that guide it toward a specific fate. Certain genes within the cell are switched on, while others are switched off. Over time, the cell begins to shape itself into the form it is destined to hold. Once the process is complete, the cell has a clear role, and its path is set. It is important to note, however, that not all stem cells carry the same degree of potential. In the earliest days of development, stem cells are pluripotent, meaning they can become any cell in the body. These embryonic stem cells are the most powerful, capable of giving rise to every tissue and organ. As development continues, many of these cells specialize, but a population remains, scattered throughout the body, where they continue to contribute to growth and repair. These later stem cells are known as adult stem cells. They are more limited, able to become only certain types of cells, yet they remain essential for maintenance and healing. One of the most well-known examples of adult stem cells can be found in the bone marrow. Here, deep within the cavities of bones, live hematopoietic stem cells. These cells are responsible for producing blood. Every day, countless red blood cells are lost as they circulate through the body, carrying oxygen to tissues. They are fragile, living only a short time before being replaced. White blood cells, the defenders of the immune system, also have limited lifespans. Without constant renewal, the body would quickly weaken. It is the stem cells in bone marrow that ensure this never happens, generating new blood cells continuously, quietly sustaining life. Another example lies in the skin. The outermost layer of skin, known as the epidermis, is in a constant state of renewal. Cells at the surface are shed every day, flaking away as part of the natural cycle of protection. Without replacement, the skin would thin and leave the body vulnerable. Stem cells located deeper within the skin ensure this cycle never ends. They divide and produce new cells, which gradually move upward, replacing the ones that are lost. In this way, stem cells are the unseen caretakers of the body's largest organ, keeping it strong, protective, and whole. The lining of the intestines offers another quiet example. The cells here face constant wear as they process the food we eat, absorbing nutrients and enduring the passage of materials. To keep this system functioning, stem cells reside in small niches along the intestinal walls. From these niches, they divide and produce fresh cells, maintaining the delicate balance of digestion and absorption. Without them, the lining of the gut would quickly erode, leaving the body unable to process nourishment. Thinking of stem cells in this way shows how they are not only the beginning of life, but also its ongoing support. They are like silent workers who never rest, ensuring that the body can continue despite the constant challenges it faces. They do not call attention to themselves, but without them, the body could not endure. The study of stem cells has revealed not only their role in daily life, but also their potential for medicine. Because stem cells can become many types of cells, scientists have long wondered whether they could be used to replace tissues damaged by injury or disease. Could new nerve cells restore movement after spinal cord injury? Could fresh heart muscle cells repair damage after a heart attack? Could insulin-producing cells help those with diabetes? These questions are not just dreams, but active areas of research. The hope is that by guiding stem cells in the right direction, we may one day harness their natural potential to heal. Yet even as we look to the future, it is worth pausing to reflect on what is already true. Stem cells are not a distant possibility. They are present now, working quietly within every person. They are in your marrow, renewing your blood. They are in your skin, maintaining its surface. They are in your gut, helping you digest. Every moment, they are there, performing their tasks without pause. This is the hidden rhythm of life, the unbroken cycle of renewal that sustains you without notice. As you imagine these cells, it can be calming to think of them as small lights scattered throughout the body. Some glow more brightly, actively dividing and producing new cells. Others glow softly, waiting for the moment when they are needed. Together, they create a quiet constellation, a network of possibility that stretches through every tissue. They remind us that life is never static, that change and renewal are always possible. In the end, stem cells are more than scientific curiosities. They are the essence of resilience. They show us that the body carries within itself the power to adapt, to recover, and to begin again. They are the building blocks not only of life's beginning, but of its continuation. A reminder that even in silence, there is strength, and even in stillness, there is possibility. 3. What are stem cells? To understand stem cells clearly, it is helpful to imagine them as the body's most versatile players, always ready to step into whatever role is needed. Most cells in the body are specialists. A heart cell beats in steady rhythm, designed to contract and relax without pause. A nerve cell carries signals, stretching across long distances to connect the brain with the farthest reaches of the body. A red blood cell carries oxygen, drifting silently through blood vessels, with a shape perfectly suited for its purpose. These cells are remarkable in their own right, but once they have committed to a task, they cannot easily change. They are dedicated to a single function. Stem cells, however, are different. They are generalists, unspecialized, open, and adaptable. They hold within themselves the capacity to become many things. A stem cell can be thought of as an unfinished blueprint. It has the basic structure, the essential materials, and the full potential to become a completed design. But it has not yet been finalized. It waits for instructions, for signals, for the environment around it to guide its fate. This is what defines stem cells. They are undifferentiated cells with two defining abilities. The first is self-renewal, the capacity to divide and create more copies of themselves. The second is differentiation, the ability to mature into specialized cells with distinct functions. These two abilities, renewal and specialization, are the hallmarks of stem cells and the reason they are so important to life. Self-renewal ensures that the supply of stem cells never runs out. When a stem cell divides, it often produces one copy of itself and one cell that is destined to become something more specific. In this way, the population of stem cells remains steady, even as they give rise to new specialized cells. It is a delicate balance, a system of preservation and contribution. Without self-renewal, the reserve of stem cells would gradually dwindle. Without differentiation, they would never fulfill their purpose. Together, these abilities create a cycle that sustains growth, development, and repair throughout life. Differentiation is the process by which a stem cell takes on a specific identity. This process is guided by both internal and external signals. Inside the cell, certain genes are activated while others are silenced. These genes produce proteins that shape the cell's structure and function. Outside the cell, chemical signals from neighboring cells, physical contact with surrounding tissues, and even the stiffness or softness of the environment all contribute to the decision. Over time, the cell gradually transforms, adopting the features and functions of a specialized cell type. A stem cell in the bone marrow, for example, may become a red blood cell, flattening into a disc shape and filling itself with hemoglobin to carry oxygen. Another may become a white blood cell, equipped with defenses to fight infection. Both began as the same stem cell, but their paths diverged based on the signals they received. Not all stem cells are the same. Their potential varies depending on where they come from and when they are found. Embryonic stem cells, discovered in the earliest stages of development, are pluripotent. This means they can become any type of cell in the body. They are the most versatile, carrying the full potential of life within them. In contrast, adult stem cells, found in specific tissues after birth, are multipotent. They can give rise only to certain types of cells related to their tissue of origin. For example, hematopoietic stem cells in the bone marrow can produce all the different kinds of blood cells, but cannot create nerve or muscle cells. This limitation reflects their specialized environment and their role in maintenance rather than total creation. A third category has emerged in recent years, known as induced pluripotent stem cells. These are adult cells that have been reprogrammed in the laboratory to return to a pluripotent state. By altering the expression of certain genes, scientists have found ways to unlock the potential of cells that had already specialized, turning them back into something more flexible. This discovery has opened new possibilities for research in medicine, as it provides pluripotent stem cells without relying on embryos. It also demonstrates the remarkable plasticity of life, the way cells can shift between states under the right conditions. Stem cells are not confined to one location in the body. They are scattered in niches, small microenvironments that protect them and regulate their activity. These niches provide signals that tell stem cells when to divide and when to remain at rest. In bone marrow, stem cells are nestled among supporting cells and blood vessels, responding to the body's need for new blood. In the skin, they rest in the basal layer, where they replace the cells that are constantly shed from the surface. In the intestines, they sit at the base of tiny folds called crypts, generating the cells that line the gut and absorb nutrients. Each niche is carefully balanced, ensuring that stem cells are neither exhausted by overuse nor wasted by inactivity. What makes stem cells so fascinating is that they embody both stability and change. They are stable in their ability to reproduce themselves, ensuring continuity across time. Yet they are also agents of change, transforming into the many different cell types that sustain life. They are both guardians of potential and engines of renewal. This dual nature gives them a unique role in biology and medicine, a role that extends from the earliest moments of development to the final days of life. It is worth pausing to reflect on how often stem cells touch our lives without notice. Every cut that heals, every strand of hair that grows, every blood cell that carries oxygen, every immune cell that fights an infection. All of these exist because stem cells quietly fulfilled their role. They do not announce themselves, yet they are indispensable. They remind us that much of life's strength lies in quiet persistence, in processes that go unseen, yet never cease. When scientists describe stem cells, they often focus on their potential for therapy and regeneration, and indeed, the possibilities are vast. Yet even without future treatments, stem cells are already working within each of us, every day. They are the reason our bodies endure, the reason wounds do not remain open, the reason blood flows fresh, and the reason the skin renews itself. They are as much a part of daily survival as they are of scientific dreams. In the simplest terms, stem cells are the body's reserve of possibility. They are the blank pages that can be written upon, the seeds that can sprout into many forms, the unfinished blueprints waiting for completion. They remind us that within every living system, there is always room for renewal, always the capacity to repair, and always the quiet power of potential. 4. The concept of differentiation. In the rhythm of life, one of the most remarkable processes is the way in which cells take on their unique identities. This process, known as differentiation, is the quiet unfolding of potential into purpose. It is the moment when a cell that was once undefined, open to all possibilities, gradually becomes something particular. A stem cell, in its earliest form, does not yet know what it will be. It floats in a state of openness, able to choose many different paths. But as time passes, as signals reach it from both within and without, it begins to narrow its possibilities. Slowly, carefully, it becomes a muscle cell, or a nerve cell, or a blood cell. Differentiation is this journey, the gentle narrowing of possibility, until a cell arrives at its destiny. To picture differentiation, one might imagine a single traveler standing at the mouth of many roads. At first, all paths are open, each leading to a different destination. The traveler waits, considering, until a signpost appears, pointing toward one road. With each step, more signposts emerge, more directions offered, more hints about where the journey leads. Eventually, the traveler reaches a place that is uniquely theirs, a place where they settle and build their life. In this way, a stem cell journeys from openness to specialization, guided not by chance, but by a complex harmony of signals. Inside the cell, The process begins with the activation of genes. Every cell in the body carries the same DNA, the same library of instructions, but not every cell reads every chapter. In a muscle cell, the chapters related to contraction are read, while others remain closed. In a nerve cell, the chapters about transmitting electrical signals are opened, while those about carrying oxygen are left untouched. Differentiation is not the addition of new instructions, but the selective reading of what is already present. It is as though each cell has access to the same vast book of knowledge, but chooses to study only the sections that pertain to its role. External signals also play a vital role. A cell does not exist in isolation. It is surrounded by other cells, by tissues, by chemical signals, by physical forces. These influences act as quiet whispers, nudging the cell toward a particular fate. Neighboring cells release molecules that drift across tiny spaces, binding to receptors and triggering changes within. The stiffness or softness of the surrounding tissue can influence whether a stem cell becomes muscle or bone. Even the flow of blood, the presence of nutrients, and the temperature of the environment can shape the decision. Differentiation is therefore not only a genetic process but also an environmental one, a conversation between the cell and its surroundings. In the earliest stages of life, when an embryo is just beginning to form, differentiation occurs on a grand scale. The pluripotent stem cells within the inner cell mass begin to diverge, some becoming the precursors of the nervous system, others the foundation of the heart, the lungs, the digestive tract, and the muscles. Layer by layer, tissue by tissue, the body takes shape. This is called embryonic differentiation, and it is the blueprint for all that follows. The same principles continue throughout life, but in smaller, quieter ways. As adult stem cells replace old cells and repair damaged tissues, differentiation is not always a one-way path. While most cells, once specialized, remain committed to their role, there are exceptions. Under certain conditions, some cells can change their identity, a process known as transdifferentiation. This is rare in nature, but scientists have learned to encourage it in the laboratory. They can take a skin cell, for example, and coax it into becoming something entirely different, such as a neuron. This flexibility shows the deep potential that lies hidden within every cell, a potential that usually remains silent but can be revealed with the right guidance. The beauty of differentiation lies not only in its mechanics but in what it represents. It shows how complexity can arise from simplicity, how unity can give way to diversity. From a single fertilized cell comes the vast array of tissues and organs that make up a human being. Each cell carries the same DNA, yet through differentiation, each becomes something distinct, contributing to the larger harmony of the body. It is a reminder that diversity of function is built upon a foundation of shared identity, a principle as true in biology as it is in life. Differentiation also reveals the balance between permanence and change. Once a stem cell commits to a particular role, it rarely goes back. A heart cell does not become a skin cell. A nerve cell does not turn into muscle. This stability ensures that tissues function reliably, that the heart continues to beat, that the brain continues to think, that the skin continues to protect. Yet the presence of stem cells ensures that the body never runs out of new recruits. While specialized cells remain fixed, stem cells remain flexible, providing a constant source of renewal. As we reflect on differentiation, it can be soothing to imagine the process unfolding quietly within the body. At every moment, stem cells are receiving signals, making decisions, and committing to paths. Some become blood cells, flowing through vessels to carry oxygen. Others become immune cells, standing guard against infection. Still others become skin cells, forming the protective layers that shield us from the world. Each one has taken a journey from openness to purpose, from potential to reality. This journey is not dramatic or sudden, but gradual, unfolding with gentle precision. In many ways, differentiation is the essence of growth. It is the process by which complexity arises from simplicity, by which the many emerge from the one. It is both an act of narrowing and an act of creation, reducing possibility while expanding function. A stem cell that becomes a neuron gives up the chance to be anything else. But in doing so, it gains the ability to transmit thought and sensation. A stem cell that becomes a red blood cell loses the ability to divide, but gains the power to carry oxygen throughout the body. Differentiation is therefore both sacrifice and fulfillment, a trade of openness for purpose. In the larger story of life, differentiation allows for cooperation. No single cell can do everything, but together, specialized cells form tissues and organs that sustain the body. Muscle cells contract, nerve cells signal, skin cells protect, blood cells transport. Each plays its role, and together they form a whole that is far greater than the sum of its parts. Differentiation makes this possible, carving distinct identities from shared beginnings. As you rest with these thoughts, you might imagine differentiation as a quiet symphony. Each stem cell begins as a musician holding a silent instrument, waiting to know what part to play. Over time, the conductor, the environment, the signals, the genes guides each musician. One begins to play a melody, another a harmony, another a rhythm. Alone, each is incomplete, but together, they create music. Differentiation is this process, the transformation of silence into song, of potential into purpose. And so, differentiation is not just a biological mechanism, but a profound reminder. It shows us how life builds complexity from simplicity, how openness gives way to identity, and how diversity can arise from unity. It is the gentle journey of a cell finding its role, the quiet unfolding of purpose from possibility, and the foundation upon which all of life's variety rests. 5. Embryonic stem cells. At the very beginning of human life, when existence is measured not in years or even days, but in the delicate divisions of a handful of cells, there is a form of potential unlike any other. Within the earliest stages of an embryo lie embryonic stem cells, cells that carry with them the vast promise of becoming every part of the human body. They are unique, unmatched in their versatility, and they remind us of how life, in its quietest origins, is filled with endless possibility. To think of embryonic stem cells is to think of a kind of openness that disappears as life becomes more defined, a moment in which the future of the body has not yet been determined, but every path remains available. Embryonic stem cells are first found in the blastocyst, a tiny structure that forms only a few days after fertilization. At this point, the embryo is no larger than a pinhead, yet within it lies a cluster of cells known as the inner cell mass. It is within this inner group that embryonic stem cells reside. These cells are pluripotent, meaning they have the capacity to become any cell type in the body. They can give rise to muscle, bone, skin, nerves, blood, or organs. No other type of stem cell carries such vast potential. While adult stem cells can only form a limited range of cells related to their tissue of origin, embryonic stem cells stand at the doorway of all possibilities. To imagine them is to picture a blank canvas before the first brushstroke. Nothing has yet been painted, but the potential for landscapes, portraits, and patterns all lie within it. Once the paint is applied, the picture takes form, and the openness narrows. In the same way, embryonic stem cells are the untouched canvas, waiting to become whatever the body requires. They are the starting point of the journey, the wellspring from which every tissue flows. The process by which these cells become specialized is both beautiful and precise. embryonic stem cells receive signals that guide them into the three primary layers of early development: the ectoderm, mesoderm, and endoderm. From these three layers, the full range of tissues emerges. The ectoderm becomes the skin and the nervous system. The mesoderm forms muscles, bones, and the circulatory system. The endoderm develops into the lining of the gut, the lungs, and many internal organs. In this way, the pluripotent stem cells of the inner cell mass gradually map out the blueprint of the body, translating possibility into form. What makes embryonic stem cells especially remarkable is not only their ability to differentiate into any cell type, but also their ability to replicate themselves indefinitely in the right conditions. In the laboratory, scientists have discovered that if these cells are provided with the proper environment, they can divide endlessly while maintaining their pluripotency. This capacity for self-renewal allows researchers to study them in detail, to observe the ways in which they respond to signals, and to experiment with guiding them towards specific fates. In this sense, embryonic stem cells are not only the foundation of life, but also a window into its mysteries. The study of embryonic stem cells has opened doors to medical research that were once unimaginable. Scientists have learned to coax these cells into forming specific types of tissues, such as heart muscle or neurons. The hope is that, in the future, such cells could be used to repair damaged organs, to replace tissue lost to injury or disease, or even to grow entirely new organs in the laboratory. The potential applications touch nearly every corner of medicine, regenerating damaged spinal cords, restoring sight to failing eyes, healing hearts after attack, or producing insulin for those with diabetes. Each of these dreams begins with the unique capacity of embryonic stem cells to become anything. But the story of embryonic stem cells is not only one of promise, it is also one of reflection and care, for their source raises questions that reach beyond science. Because embryonic stem cells are derived from early embryos, their use has sparked ethical debates about the beginning of life, about what should and should not be done in pursuit of healing. For some, the potential to save lives and reduce suffering outweighs the concerns. For others, the origins of these cells make their use deeply problematic. This ongoing dialogue has led to new approaches, such as the development of induced pluripotent stem cells, which allow scientists to reprogram adult cells into a pluripotent state without relying on embryos. In this way, the science of stem cells has evolved not only through discovery but also through reflection, adapting to balance potential with principle. Yet beyond the ethical debates, embryonic stem cells remain a profound reminder of the flexibility and resilience of life. They embody a moment of openness, a fleeting stage in which all futures are possible. As the embryo develops, that openness gradually narrows. Cells commit to paths, tissues take shape, and the body begins to form. The pluripotency of embryonic stem cells is therefore not a permanent state but a temporary gift, one that lays the foundation for everything that follows. It is calming to think of embryonic stem cells as the first architects of the body. With quiet precision, they begin the construction of bones, organs, and systems, setting the stage for growth and survival. They are unseen, their work silent, yet their influence touches every aspect of life. Every heartbeat, every breath, every step taken later in life is possible because embryonic stem cells once divided, once differentiated, once set the path in motion. In the laboratory, when scientists observe embryonic stem cells under the microscope, they often see clusters of small, rounded cells, glowing softly in their cultures. To the eye, they look simple, almost unremarkable, yet within them lies the ability to generate entire organisms, to form the complexity of life from a blank beginning. This duality appearance of simplicity, possession of infinite potential, is what makes them so compelling. They remind us that greatness often begins in quiet, unassuming places. As you let your thoughts linger on these cells, you may imagine them as stars in the night sky, each one capable of shining into a different form. Some will become the glow of skin, others the fire of muscle, others the delicate sparkle of neurons. Together, they create the constellation of the human body, each point of light contributing to the whole. The journey from stem cell to specialized cell is the journey from possibility to reality. And embryonic stem cells mark the first step. In the gentle story of life, embryonic stem cells are the opening chapter. They are the beginning, the source of all potential, the moment in which the body is still a dream waiting to take shape. They are the reminder that every complex form, every movement, every thought begins from something small, quiet, and open to everything. To reflect on them is to reflect on the beauty of beginnings, the mystery of potential, and the calm unfolding of life from its earliest whisper. 6. Adult stem cells. As life progresses beyond its earliest moments, the story of stem cells does not end. While embryonic stem cells shape the first chapters of existence, a different kind of stem cell remains behind, carried with us into birth, into growth, and into adulthood. These are adult stem cells, sometimes called somatic stem cells. They are not as versatile as their embryonic counterparts, but they are steady, reliable, and essential. They serve as the caretakers of the body, quietly repairing, renewing, and replacing cells as needed. Without them, the body would not be able to sustain itself over time, for tissues wear down, blood cells age, skin sheds, and injuries demand repair. Adult stem cells make sure that the cycle continues, day after day, year after year. To imagine adult stem cells is to picture a small reserve of potential hidden within the body's tissues. They are not scattered everywhere, but instead reside in carefully chosen niches, environments designed to protect them and regulate their activity. These niches act like shelters, keeping the stem cells safe, while also providing them with signals that guide when they should remain at rest and when they should awaken. Within bone marrow, for instance, hematopoietic stem cells lie nestled among supporting cells and blood vessels. They wait there, quiet and still, until the body calls upon them to produce new blood. When that call comes, they divide and differentiate, generating the red blood cells that carry oxygen, the white blood cells that defend against infection, and the platelets that help blood clot. Each of these vital elements of blood begins as an adult stem cell, transformed through a process as natural as breathing. The skin offers another clear example. The surface of the skin, constantly exposed to the outside world, wears down quickly. Cells at the outermost layer die and are shed, replaced by newer cells moving up from beneath. If no mechanism existed to replace these cells, the protective barrier of the skin would erode, leaving the body vulnerable. But stem cells dwelling deep in the basal layer of the skin take care of this task. They divide, producing daughter cells that move upward, gradually flattening and toughening as they reach the surface. In this way, adult stem cells ensure that the skin renews itself continuously, maintaining its role as a shield against the world. The lining of the gut provides yet another example of the quiet diligence of adult stem cells. The intestinal lining is one of the fastest renewing tissues in the body, replaced every few days. Stem cells located in the crypts, the small folds at the base of the intestinal lining, divide constantly to generate fresh cells. These new cells move upward, absorbing nutrients and aiding in digestion until they eventually wear out and are replaced by the next generation. It is a cycle that never pauses, driven by stem cells working in silence. Compared with embryonic stem cells, adult stem cells are more limited in their potential. They are multipotent rather than pluripotent, able to form only certain types of cells. A hematopoietic stem cell can give rise to all the varieties of blood cells, but it cannot transform into a nerve or a muscle. A neural stem cell in the brain can produce neurons in supporting glial cells, but it cannot produce red blood cells or skin. This limitation reflects their purpose. While embryonic stem cells create the full architecture of the body, adult stem cells maintain and repair specific structures that already exist. They are specialists in renewal, tied to the tissues they protect. Despite these limitations, adult stem cells are indispensable. They allow the body to survive injuries, to adapt to wear and tear, and to replace what has been lost. Consider a cut on the skin. Without stem cells, the wound would remain open, the barrier broken. With them, the gap closes as new skin cells form. Consider the constant loss of blood cells. Without stem cells, oxygen delivery would falter, and the immune system would collapse. With them, the supply is steady and unbroken. Adult stem cells ensure that the body does not exhaust itself, that life continues despite the passage of time. The discovery of adult stem cells has transformed medicine as well. The earliest practical use came in the form of bone marrow transplants. In cases of leukemia or other blood disorders, diseased marrow can be replaced with healthy marrow from a donor. What makes this possible is not the marrow itself, but the stem cells within it. These transplanted stem cells find their way into the recipient's bones, settle into new niches, and begin producing healthy blood. For many patients, this procedure has meant the difference between life and death. It is one of the earliest examples of stem cells used in therapy, and it remains a cornerstone of medical practice today. In recent years, scientists have discovered adult stem cells in places once thought unlikely. Neural stem cells have been found in certain regions of the brain, offering hope that damaged nervous tissue may one day be repaired. Stem cells in muscle help regenerate fibers after injury, while others in the liver contribute to the organ's remarkable ability to recover from damage. Even fat tissue, once thought to serve only as storage, has revealed populations of stem cells that may have therapeutic potential. These discoveries expand our understanding of the body's quiet resilience, showing that stem cells are more widespread and more versatile than once believed. What makes adult stem cells so fascinating is not only their role in renewal, but also the way they embody balance. They must divide enough to maintain tissues, but not so much that they overwhelm them. They must remain stable across decades, avoiding exhaustion or error. This delicate balance is maintained by the niches that house them, the environments that protect them and provide the right signals. When the balance falters, disease can arise. Too little renewal leads to degeneration, as tissues weaken and fail. Too much uncontrolled renewal can lead to cancer, as stem cells divide without restraint. The harmony of adult stem cells is therefore essential to the harmony of the body itself. It is calming to reflect on the way adult stem cells serve us quietly, invisibly, throughout our lives. Every day, without notice, they replace millions of cells, patching the body where it has worn thin, refreshing it where it has grown old. They are not celebrated, but they are always present, like unseen gardeners tending the landscape of the body. They prune, they replenish, they restore, ensuring that life continues with gentle persistence. When one thinks of adult stem cells, one sees not the boundless possibility of embryonic cells, but the steady endurance of maintenance and care. They are not the open canvas of beginnings, but the careful brushstrokes that preserve the painting once it has been made. They are the quiet companions of life, always at work, always ensuring continuity. Without them, existence would be brief and fragile. With them, it is sustained and resilient. And so, in the larger story of stem cells, adult stem cells are the keepers of the present. They do not shape the first moments of life, nor do they carry the infinite promise of becoming everything. Instead, they hold the gift of renewal within the context of what already island. They ensure that the structures built in the beginning do not fall apart, that the body remains whole and functional across the years. They are the guardians of continuity, the gentle workers who preserve the body's harmony. To reflect on them is to reflect on the endurance of life itself, the quiet strength that sustains us from one moment to the next. 7. Induced pluripotent stem cells. In the long story of stem cell research, there is a chapter that feels almost like a return to beginnings. For many years, scientists believed that once a cell had chosen its path, once it had become muscle or skin or blood, it could never go back. Differentiation was seen as a one-way journey, a road that led from openness to purpose with no possibility of reversal. A skin cell was destined to remain a skin cell, a nerve cell destined to remain a nerve cell. But in the early years of the 21st century, researchers discovered something that changed this understanding forever. They found that with the right signals, an adult cell could be reprogrammed to return to a state very much like that of an embryonic stem cell. These reprogrammed cells are called induced pluripotent stem cells, or iPSCs, and they represent one of the most remarkable discoveries in modern biology. To imagine this process, picture a book that has already been written and published, its story fixed, and its pages filled. What researchers discovered was a way to erase the ink, to restore the pages to blankness, and to allow the story to be written again. A skin cell, which had already committed itself to forming the body's protective surface, could be guided back into a pluripotent state, able once more to become anything: heart cell, neuron, blood cell, or muscle. In this way, iPSCs mirror the abilities of embryonic stem cells without coming from embryos. They are the rediscovery of possibility within cells that had seemed forever specialized. The process of creating iPSCs involves carefully introducing specific genes into adult cells. These genes act as master regulators, unlocking the portions of DNA that had been closed off during differentiation. When these regulators are activated, they erase the cell's commitment and restore its openness. The cell, once fixed in its role, becomes pluripotent again, able to divide endlessly and to differentiate into many types of cells. It is as though the cell has been invited to step back in time, returning to a moment when its future was still unwritten. The significance of this discovery cannot be overstated. Before iPSCs, much of the promise of stem cell research depended on embryonic stem cells. While powerful, their use raised ethical questions, since they were derived from early embryos. With iPSCs, scientists found an alternative, a way to obtain pluripotent cells without relying on embryos. This breakthrough not only sidestepped ethical concerns but also opened the door to new possibilities. Because iPSCs can be created from a patient's own cells, they offer the potential for therapies that are perfectly matched, reducing the risk of rejection. They also provide a model for studying diseases in ways that were previously impossible. Imagine, for example, a patient with a genetic disorder. Scientists can take a small sample of skin cells from that patient, reprogram them into iPSCs, and then guide them to become the specific cell type affected by the disease. In this way, researchers can study the condition in the laboratory, observing how the disease unfolds at the cellular level. They can test treatments directly on these cells, tailored to the individual. This approach has already provided insights into conditions such as Parkinson's disease, heart disorders, and certain types of diabetes. It allows science to move beyond generalized study and into the realm of personalized medicine. Another promise of iPSCs lies in regenerative therapy. Because they can become many types of cells, they may one day be used to repair tissues damaged by injury or illness. A person who has lost heart muscle after a heart attack might receive new heart cells grown from their own skin, reprogrammed into iPSCs, and then guided toward a cardiac fate. Someone with spinal cord injury might benefit from neurons grown in the same way. The dream is that the body's own cells, returned to their earliest state, can be used to repair what has been lost. Even now, researchers are learning how to refine the process, making it safer and more efficient. Early methods of creating iPSCs involved introducing genes using viruses, a technique that carried risks. Newer approaches use chemical signals or precise editing tools, offering greater control. The work is delicate, requiring patience and precision, but with each advancement, the potential grows clearer. Beyond therapy, iPSCs also help us understand the nature of life itself. They demonstrate that identity within the cell is not as rigid as once believed. A cell's fate can be altered, its path reversed, its openness restored. This flexibility suggests that life carries within it a resilience deeper than we imagined, a capacity for renewal that extends even to the level of individual cells. It is a reminder that biology is not fixed but dynamic, not closed but always shifting. It is calming to think of iPSCs as a bridge between beginnings and continuations. Embryonic stem cells represent the earliest openness of life, a fleeting moment of possibility before specialization begins. Adult stem cells represent maintenance, the steady renewal of tissues within the boundaries of their origins. Induced pluripotent stem cells stand between these two states, a return to beginnings drawn from the middle of life. They show us that even cells that have chosen a path can find their way back to openness. That potential can be rediscovered even after years of specialization. As you reflect on iPSCs, you might imagine them as doors reopening after long being closed. A skin cell, once certain of its role, finds itself with a key, unlocking pathways to many futures. A blood cell, once fixed in its duties, is invited to step back and choose again. This imagery captures the essence of iPSCs. They are the restoration of choice, the renewal of possibility, the quiet rewriting of fate. The discovery of iPSCs also highlights the deep interplay between knowledge and imagination. Scientists had long wondered whether differentiation could be reversed, whether cells could be coaxed back into openness. For years, it seemed impossible. But persistence and creativity revealed that the answer was yes. This breakthrough reminds us that biology is not only a field of observation but also of exploration, where curiosity can uncover truths hidden within the fabric of life. In the end, induced pluripotent stem cells represent more than just a scientific achievement. They symbolize renewal, the possibility of returning to beginnings, the idea that even within the fixed paths of life, there may be ways to rediscover openness. They are a quiet reminder that change is always possible, that potential can be regained, and that within the smallest parts of ourselves lies a capacity for transformation. To rest with these thoughts is to rest with the comfort that life is not a closed story, but an unfolding one, with new beginnings always waiting to emerge. 8. Stem cells in blood. Among all the places where stem cells quietly work within the body, few are as vital as the blood. Blood is the river of life, flowing constantly through veins and arteries, carrying with it oxygen, nutrients, hormones, and the many signals that allow the body to function as one. Yet this river is not a static fluid. It is alive, dynamic, always in motion, and always being replenished. Every drop of blood contains countless cells, each performing a specific duty, and all of these cells have limited lifespans. Red blood cells, for instance, circulate for only a few months before they wear out and are removed from the body. White blood cells, the guardians of the immune system, may last only days or weeks, depending on their type. Platelets, the fragments that help blood clot, survive for just over a week. Without renewal, the blood would quickly lose its function, and life would fade. It is here that stem cells, particularly hematopoietic stem cells, play their most constant and essential role. Hematopoietic stem cells, often referred to as HSCs, reside in the soft tissue inside bones known as bone marrow. Hidden away in these protective spaces, they serve as the fountainhead from which all blood cells arise. They are multipotent, meaning they cannot become every cell type in the body, but they can produce every variety of blood cell, both the red cells that transport oxygen and the many forms of white cells that defend against infection. They can also give rise to platelets, ensuring that blood can clot when a vessel is damaged. In this way, HSCs are responsible for the ceaseless renewal of the body's most fluid tissue. The process by which HSCs maintain the blood is both intricate and balanced. When an HSC divides, it creates two daughter cells. One often remains a stem cell, preserving the pool for future use. The other takes a different path, gradually moving down the road of differentiation. Along this path, the cell commits to one of two major lineages: the myeloid lineage or the lymphoid lineage. From the myeloid side come red blood cells, platelets, and several types of white blood cells, including neutrophils and monocytes. From the lymphoid side arise lymphocytes, such as T cells, B cells, and natural killer cells, each with a specialized role in defending the body. Through this branching system, HSCs ensure that every type of blood cell is produced in the right numbers, at the right time, to meet the body's needs. The scale of this renewal is astonishing. Every day, the human body produces billions of new blood cells. Each of these cells begins as a stem cell, shaped by signals within the marrow. The demand is constant and unrelenting, for the circulation of blood requires a steady supply. If red blood cells are not replenished, tissues would be starved of oxygen. If white blood cells decline, the body would lose its ability to resist infection. If platelets were missing, even small wounds would lead to dangerous bleeding. The quiet activity of HSCs in the marrow prevents these crises, keeping the blood alive and functional. The importance of these cells becomes especially clear in times of stress. When the body is injured, when infection takes hold, or when bleeding occurs, the demand for certain blood cells rises sharply. HSCs respond by increasing production. In the presence of infection, for example, chemical signals from immune cells travel to the marrow, stimulating the rapid production of white blood cells. In cases of blood loss, signals trigger the production of red blood cells and platelets. The system is flexible, always adjusting to maintain balance, always responding to the body's needs. The role of HSCs in health is matched by their importance in medicine. Bone marrow transplantation, a procedure that has saved countless lives, depends entirely on these cells. In conditions such as leukemia, where the marrow itself produces abnormal or cancerous blood cells, the only hope may be to replace the diseased marrow with healthy marrow from a donor. The key to this treatment is the transplantation of HSCs. When infused into a patient's bloodstream, these stem cells travel to the bones, settle into new niches, and begin the work of producing healthy blood. It is a remarkable process in which a new population of stem cells takes root and restores what was lost. Beyond transplantation, HSCs are also central to the study of many diseases. Because they give rise to all blood cells, they provide a model for understanding disorders of immunity, clotting, and oxygen transport. Scientists can study how HSCs respond to genetic mutations, infections, or treatments, gaining insight into conditions that affect millions. They also serve as a testing ground for new therapies, allowing researchers to explore ways to correct defects or enhance function. It is calming to think of HSCs as guardians of the bloodstream, constantly at work beneath the surface, ensuring that the body never runs dry of its essential elements. They do not move through the vessels themselves, but remain rooted in the marrow, like springs feeding a river. From these hidden springs, the blood is renewed, its flow unbroken. its vitality sustained. Every heartbeat, every breath, every moment of life depends on their quiet persistence. The imagery of the marrow as a hidden garden is fitting. Within its soft spaces, HSCs are nurtured, protected, and guided. The niches in which they reside provide not only shelter but also the signals that regulate their activity. Certain supporting cells in the marrow release molecules that tell HSCs when to divide, when to remain at rest, and when to differentiate. This balance ensures that the supply of stem cells is preserved, even as they generate new blood cells. Too much division would exhaust the pool, while too little would leave the blood depleted. The niche ensures harmony, a delicate equilibrium that sustains life. Even as we understand their function, there remains something profoundly comforting about the idea of stem cells in the blood. They remind us that renewal is not occasional but constant, that the body carries within itself the ability to refresh its most essential fluid every moment of every day. They are a symbol of resilience, a reminder that even as cells die, new ones are born, and the flow of life continues without interruption. To reflect on HSCs is to reflect on the ceaseless cycle of life within. Red blood cells carrying oxygen, white cells fighting infection, platelets sealing wounds. All of these are temporary, each with its own lifespan. But the stem cells in the marrow endure, generation after generation, ensuring that the river of blood never runs dry. They are not visible, yet they are always present, unseen but indispensable. They are the silent workers of the bloodstream, the foundation upon which circulation depends. In the end, stem cells in the blood are not only a scientific marvel, but also a gentle lesson in continuity. They show us that life persists through constant renewal, that loss is balanced by creation, and that the flow of vitality is sustained by the quiet work of cells we never see. They are the springs that feed the river, the roots that sustain the forest, the unseen presence that makes every heartbeat possible. To think of them is to be reminded of the quiet strength that lies within, the resilience that flows through every vein, and the calm truth that renewal is always happening, even in silence. 9. Stem cells in the brain. The brain is often thought of as the seat of identity, the organ that allows us to think, to remember, to feel, and to dream. It is a structure of immense complexity, composed of billions of neurons connected by trillions of synapses, forming networks that create consciousness itself. For much of history, it was believed that this organ was fixed once formed. Neurons, it was thought, could not be replaced once they were lost. The brain was considered a static structure, able to adapt through the strength of its connections but unable to regenerate. Yet, in the quiet discoveries of recent decades, scientists have found that this is not entirely true. Hidden within certain regions of the brain are populations of stem cells, neural stem cells, that continue to divide and give rise to new neurons and supporting cells throughout life. Though limited in number and activity, these cells reveal that even the most intricate of organs retains a capacity for renewal. To understand neural stem cells, it helps to step back to the earliest stages of brain development. In the embryo, the nervous system begins as a simple sheet of cells that folds and closes to form the neural tube. Along this tube, stem cells proliferate, producing the first neurons and glial cells that will populate the brain and spinal cord. These embryonic neural stem cells are the architects of the nervous system, dividing and differentiating to create the diverse array of cell types required for function. As development progresses, most of these stem cells commit to specialized roles, forming the vast architecture of the brain. Yet a small number remain behind, preserved in niches where they continue to serve into adulthood. In the adult brain, neural stem cells are found primarily in two regions: the subventricular zone, which lines the lateral ventricles deep within the brain, and the hippocampus, a structure involved in memory and learning. In these niches, stem cells rest quietly, dividing occasionally to produce new neurons or glial cells. The process is far slower and more limited than in the developing embryo, but it is nonetheless remarkable. It means that the brain, once thought to be static, is in fact dynamic, capable of generating new cells, even in maturity. The hippocampus is particularly important in this regard. It plays a central role in the formation of new memories, in learning, and in spatial navigation. Within the hippocampus lies the dentate gyrus, where neural stem cells give rise to new neurons throughout life. These newborn neurons gradually integrate into existing circuits, becoming part of the networks that encode experience and knowledge. Research suggests that this process, known as adult neurogenesis, contributes to the flexibility of memory and the ability to adapt to new situations. It may also play a role in emotional regulation, helping the brain respond to stress and mood changes. The subventricular zone provides another example of ongoing renewal. Here, neural stem cells produce progenitor cells that migrate along a pathway to the olfactory bulb, a structure involved in the sense of smell. In humans, this process appears to be less pronounced than in other animals, but it demonstrates that the brain retains a quiet capacity for self-renewal. Even in the most protected and complex of organs, stem cells continue their work, hidden from view but essential to the balance of function. The existence of neural stem cells has profound implications. It suggests that the brain is not as fragile as once believed, that it has within it the tools to adapt and repair, at least to some extent. This has inspired new lines of research aimed at understanding how these cells can be guided or enhanced. Could stimulating neural stem cells help repair damage after a stroke? Could encouraging their activity restore memory in neurodegenerative diseases, such as Alzheimer's? Could they be used to replace neurons lost in conditions like Parkinson's disease? These questions remain open, but the presence of neural stem cells makes them possible to ask. It is calming to think of these cells as quiet gardeners within the brain, tending small plots of renewal in a vast landscape of thought. They are not abundant, nor do they replace every neuron that is lost, but their presence reminds us that the brain is never fully fixed, that even in maturity, there is room for growth and change. The networks of the brain are not only strengthened by experience, but refreshed by the addition of new participants, cells born from stem cells and integrated into the whole. The story of neural stem cells also highlights the delicate balance of their activity. Too little renewal can contribute to decline, as in aging or disease, when the birth of new neurons slows. Too much uncontrolled renewal can lead to problems as well, such as the formation of tumors. The niches where these stem cells reside regulate their activity carefully, ensuring that the right number of new cells is produced at the right time. It is a harmony of rest and activity, preservation and contribution, maintained across decades of life. There is also a poetic quality in the idea that the hippocampus, the structure most associated with memory, is one of the few regions where new neurons are born. Memory itself is the record of change, the imprint of experience upon the brain. That this structure should also be a site of renewal feels fitting, as though memory and growth are intertwined. Each new neuron that joins the network may carry with it the possibility of new learning, new adaptation, new resilience. In reflecting on neural stem cells, one might imagine the brain as a vast forest. Most of the trees are old, established, rooted firmly in place. They form the structure of the forest, providing stability and strength. But in certain clearings, small saplings continue to grow, rising from seeds that had waited quietly in the soil. These saplings do not replace the entire forest, but they bring freshness, flexibility, and new life. Neural stem cells are those seeds, sprouting quietly in hidden corners, ensuring that the forest of the brain remains alive and dynamic. The discovery of neural stem cells challenges the old view of the brain as a static organ. It shows us that life's capacity for renewal extends even to the most intricate systems. It reminds us that growth is not limited to youth, that change is always possible, and that the brain carries within itself the potential for adaptation across the lifespan. These cells may be few, their work subtle, but their presence is profound. They embody the resilience of the mind, the ability to continue learning, adapting, and remembering even as years pass. And so, stem cells in the brain are more than a scientific curiosity. They are a symbol of hope, a reminder that renewal is always possible, even in the most complex and precious of organs. They tell us that the brain is not fixed but fluid, not closed but open, always capable of quiet growth. To think of them is to rest with the comfort that within the vast networks of thought, there are always new beginnings, small and silent, waiting to take their place. 10. Stem cells in skin and gut. The surface of the body and the lining of the gut are two of the busiest frontiers of human life. They are the places where the body meets the outside world, where constant contact, friction, and exposure wear away at living tissue. And yet, day after day, these tissues remain intact, resilient, and renewed. This continuity is possible only because of stem cells, quietly working in the background to replace what is lost. Skin and gut stem cells form two of the most active populations of adult stem cells, their work unending, their rhythm constant. They are silent custodians, guarding the body's boundaries with diligence and care. The skin is the largest organ of the human body, stretching across its entire surface as a protective barrier. Every moment, it shields us from sunlight, pathogens, toxins, and physical abrasion. Yet, despite its resilience, the outermost layer of skin is fragile and temporary. Cells at the surface, known as keratinocytes, live only a short time before they dry, harden, and are shed. This process is visible in the small flakes that fall away daily, unnoticed but constant. Without replacement, the skin would quickly thin, and its protective function would collapse. To prevent this, stem cells in the basal layer of the epidermis divide steadily, producing new keratinocytes that rise to the surface. In a matter of weeks, a cell born deep within the epidermis reaches the top layer, where it finishes its life as part of the barrier. The cycle repeats endlessly, a quiet rhythm that keeps the skin whole. These skin stem cells are not scattered randomly, but organized into niches, particularly around the base of hair follicles and within the deepest layer of the epidermis. Each niche is like a workshop where stem cells wait for signals. Some divide to replace themselves, ensuring the niche remains stocked. Others divide to produce cells destined for the surface. The balance of self-renewal and differentiation ensures that the skin never runs out of cells, no matter how often it is worn away. It is this constant renewal that allows wounds to heal, scratches to close, and burns to be repaired. When the skin is injured, stem cells near the site are activated, producing large numbers of new cells that migrate to cover the wound. Their quiet presence turns injury into recovery. The skin's stem cells are also adaptable. Under normal conditions, they produce the keratinocytes that form the barrier. But under stress, such as after injury, they can produce other types of cells needed for repair, including those that rebuild hair follicles or glands. This flexibility makes them indispensable to survival. They are the reason the skin, despite its daily losses, remains a constant shield, refreshed and renewed. If the skin is the body's outermost defense, the gut is its most inward one. The lining of the intestine is another frontier, facing the constant passage of food, fluids, and microorganisms. Unlike the skin, which loses its surface gradually, the cells of the gut lining are replaced even more rapidly. The entire lining of the intestine renews itself every few days, one of the fastest rates of turnover in the body. This extraordinary pace is necessary because the gut must remain intact to digest food, absorb nutrients, and block harmful substances. Without constant renewal, the lining would erode and digestion would fail. The key to this process lies in small structures called crypts, tiny folds at the base of the intestinal lining. Within each crypt are stem cells, nestled safely in a niche that protects them and provides the signals they need. These intestinal stem cells divide rapidly, producing daughter cells that migrate upward along the sides of the crypt. As they rise, they begin to differentiate, becoming the many types of specialized cells that line the gut. Some become absorptive cells, drawing nutrients from food. Others become goblet cells, secreting mucus to protect the lining. Still others become enteroendocrine cells, releasing hormones that regulate digestion. By the time these cells reach the top of the crypt, they are fully mature, performing their functions for a short time before being shed into the intestinal cavity. The process is seamless, a continuous conveyor of renewal. The intestinal stem cells are regulated by a delicate interplay of signals. Neighboring cells release molecules that keep the stem cells active, while others provide cues that direct differentiation. This ensures that the right balance of cell types is produced. Too few absorptive cells would starve the body of nutrients. Too few goblet cells would leave the lining unprotected. Too much or too little renewal could cause disease. The niche maintains harmony, guiding the stem cells with precision. What makes the gut especially remarkable is the speed and scale of this renewal. Every day, millions of cells are shed and replaced, a rhythm so fast that the entire lining is new within a week. This constant turnover protects the body, ensuring that damaged or infected cells are quickly removed and replaced. It also allows the gut to adapt to changes in diet, environment, and microbial populations. The intestinal stem cells are the heart of this adaptability, responding with flexibility to the body's needs. Both skin and gut stem cells illustrate the quiet resilience of the body. They are not dramatic or rare, but ordinary, constant, and reliable. They remind us that survival is not only about grand events, but about steady renewal, about the countless small acts of replacement that keep the body whole. Every scratch that heals on the skin, every meal that is digested in the gut, is made possible by these cells. The presence of stem cells in skin and gut also highlights a deeper truth, that life is built on cycles of loss and renewal. Cells die, wear away, and fall aside, but new ones are always rising to take their place. This cycle is not a flaw but a strength. It ensures that tissues remain fresh, that damage is repaired, that the body is never static. It is a reminder that impermanence is not weakness, but the foundation of resilience. Scientists studying these stem cells have also begun to explore their potential in medicine. Skin stem cells have been used in grafts to treat burns, providing new layers of protection for patients who have lost their own. Intestinal stem cells are being studied for their role in diseases such as Crohn's disease and ulcerative colitis, conditions in which renewal is disrupted. Understanding how these stem cells work may open doors to therapies that restore balance, healing tissues that struggle to repair themselves. It is calming to reflect on the work of these stem cells, to imagine them laboring quietly beneath the surface. In the skin, they are the reason wounds close, the reason surfaces remain smooth, the reason the barrier holds. In the gut, they are the reason food is absorbed, the reason nutrients nourish, the reason the lining stays intact. Their rhythm is steady, their work unending, their presence essential. They are reminders that renewal is always happening, even when unseen. In the end, stem cells in the skin and gut are symbols of continuity. They show us that the body is never still, that it is always turning over, always remaking itself. They teach us that resilience is not about never being worn down, but about always being renewed. They are the quiet workers of the body's boundaries, sustaining the surfaces that connect us to the world and the linings that protect us within. To rest with these thoughts is to rest with the comfort that life's frontiers are always guarded, always repaired, and always refreshed by the steady hands of stem cells. 11. The healing role of stem cells. The human body is remarkable in its ability to endure, to adapt, and to recover from the many injuries and stresses it encounters throughout life. A scraped knee, a broken bone, a cut on the finger, or even the strain of daily use all require some degree of repair. Much of this healing takes place so quietly that we hardly notice it. Wounds close, bones knit together, and tissues that seem damaged begin to function again. At the heart of these processes are stem cells, the body's quiet healers, responding with patience and precision whenever injury occurs. They are not dramatic in their work, nor are they noticed in the moment, but without them, healing would not be possible. When injury strikes, the body launches a coordinated response. Blood rushes to the site, clotting factors work to seal the wound, and immune cells arrive to clear away debris. Yet the true repair comes later, when new cells are needed to replace those that have been lost. This is where stem cells step in. Resting in their niches throughout the body, they sense the signals released by damaged tissues. Chemical messengers call them forth, activating their division and guiding their differentiation. A skin stem cell, for example, will begin producing keratinocytes to close a wound. A bone stem cell will generate osteoblasts to rebuild a fracture. A muscle stem cell will divide into myoblasts that fuse together, repairing torn fibers. Each tissue has its own population of stem cells, and each responds in its own way. But the principle is always the same. Stem cells awaken to restore what has been broken. One of the clearest examples of this process occurs in bone. Bones are strong, but they are not unbreakable. When a fracture occurs, stem cells in the surrounding tissue are activated. They generate new bone-forming cells that gradually bridge the gap, depositing minerals and rebuilding the structure. Over time, the bone regains its strength, often becoming even stronger at the site of the break. Without these stem cells, fractures would remain permanent, leaving the skeleton fragile and incomplete. With them, the body regains its integrity, continuing its work as if nothing had been lost. The skin offers another familiar example. Every scrape or cut is a small injury to the body's barrier, leaving it vulnerable to infection. The initial clotting of blood forms a temporary patch, but true healing requires stem cells. From the basal layer of the epidermis and from hair follicles nearby, stem cells divide rapidly, sending new cells to migrate across the wound. They knit the edges together, replacing the damaged tissue with fresh layers. Eventually, the wound closes completely, leaving behind only a faint scar, or sometimes no mark at all. This everyday miracle is made possible by stem cells, whose constant readiness ensures that the skin never remains broken for long. Muscle, too, relies on stem cells for healing. Embedded along the fibers of skeletal muscle are satellite cells, a type of muscle stem cell that remains quiet until called upon. When fibers are torn through exercise, injury, or strain, these cells activate. They divide and fuse with damaged fibers, restoring their strength and function. In this way, muscles recover from exertion and grow stronger with use. The soreness after physical activity, followed by gradual recovery, is a process shaped by these stem cells working invisibly to repair the wear and tear of movement. Even the heart, once thought to be incapable of healing, has shown some degree of regenerative capacity. Though limited, populations of cardiac stem cells and progenitor cells appear to contribute to the replacement of heart muscle after injury. Scientists are still working to understand how this process works and how it might be enhanced. The possibility that the heart could be guided to heal itself more fully offers hope for the treatment of conditions such as heart attacks, where the loss of muscle has long been considered irreversible. Beyond physical injury, stem cells also play a role in the immune system's ability to recover from illness. When an infection strikes, white blood cells are consumed in the battle to defend the body. Stem cells in the bone marrow replenish these cells, ensuring that the immune system never exhausts itself. Even after severe infections, when the immune response is depleted, hematopoietic stem cells restore the balance, preparing the body for the next challenge. In this way, stem cells are not only healers of tissue but also of defenses, constantly renewing the army that protects us from harm. The healing role of stem cells is not always complete. In some tissues, their ability to regenerate is strong, as in the skin, the blood, and the gut. In others, such as the brain and the heart, their capacity is far more limited. Yet, even here, the presence of stem cells provides a foundation for recovery, however small. Scientists are working to understand why some tissues regenerate more fully than others, and how stem cells might be encouraged to do more. The hope is that by learning from nature's own healing processes, medicine can extend their power to restore even the most stubborn injuries. There is a gentle poetry in the way stem cells respond to injury. They do not rush blindly to every signal but act with precision, dividing just enough to replace what is lost, differentiating into exactly the cells required. They are efficient, restrained, and purposeful. Their work is quiet, happening beneath the surface, unseen by the person they serve. Yet the results are clear: wounds that close, bones that mend, tissues that regain their strength. To reflect on their role is to recognize that healing is not a single event but a process, guided by the steady hands of cells that never cease their labor. The healing power of stem cells also extends to the promise of future therapies. If stem cells already repair tissues naturally, perhaps they can be guided to do even more. Scientists are exploring ways to transplant stem cells into damaged tissues, to amplify their presence where the body's own supply is too limited. Experiments with bone marrow stem cells, muscle stem cells, and even neural stem cells are beginning to show that it may be possible to enhance healing, to regenerate tissues once thought irreparable. These efforts build upon the quiet truth that stem cells are already healers, simply waiting to be directed. It is soothing to imagine stem cells as small lanterns glowing within the body, scattered in niches, each ready to light up when the darkness of injury falls. Some remain dormant for years, until the right signal awakens them. Others are constantly active, like the stem cells of the blood and skin, always renewing, always repairing. Together, they form a system of guardianship, ensuring that the body endures, despite the inevitability of damage. In the end, the healing role of stem cells is a reminder that resilience is built into life itself. The body does not merely endure injury, it responds, repairs, and restores. Stem cells are the agents of this resilience, the quiet workers that transform damage into recovery. They remind us that healing is not a miracle performed from outside, but a process woven into the very fabric of our being. To rest with this thought is to rest with the knowledge that renewal is always possible, that the body carries within it the power to mend, and that life is shaped not only by what is broken, but by the quiet forces that restore it again and again. 12. Stem cells in medical therapy. When scientists first uncovered the unique qualities of stem cells, it was clear that these remarkable cells might one day be used to heal in ways no other treatment could achieve. Over time, research has revealed not only the promise of stem cells, but also their practical applications in medicine. Some of these applications are already in use, saving lives and restoring health every day. Others are still being explored, moving slowly from the laboratory toward the clinic. Together, they form a picture of medicine shaped by the quiet potential of cells that can renew and transform. Stem cells in medical therapy represent both the present reality of treatment and the future horizon of healing. The earliest and most well-established use of stem cells in medicine comes from bone marrow transplants. This therapy has been used for decades to treat diseases of the blood, particularly leukemia, lymphoma, and certain forms of anemia. In these conditions, the body's own hematopoietic stem cells are either destroyed by disease or malfunction in ways that threaten life. By transplanting healthy bone marrow, and with it healthy stem cells, doctors can restore the production of normal blood cells. The transplanted stem cells migrate to the recipient's bone marrow, settle into new niches, and begin producing fresh blood. For many patients, this therapy has meant a chance at survival, a renewal of life through the quiet work of stem cells. Stem cells are also used in the treatment of immune system disorders. Certain inherited conditions leave patients without functional immune defenses, leaving them vulnerable to infections that most people easily resist. Bone marrow or cord blood transplants can provide these patients with new hematopoietic stem cells capable of building a functioning immune system. This approach has restored health to children born with severe combined immunodeficiency, a condition once considered fatal. The gift of stem cells, given through transplant, becomes the gift of a working immune system, offering protection where there was once none. Beyond the blood and immune system, stem cells have also been used in therapies for skin. In cases of severe burns, when large areas of skin are destroyed, doctors can use stem cells to grow new sheets of skin for grafting. These grafts are created from a patient's own stem cells or from donor sources, and are placed over the injured area to restore the protective barrier. This approach has transformed burn treatment, turning injuries that once carried little hope into cases where recovery and survival are possible. The skin's natural ability to renew itself has been amplified by medical science, guided by an understanding of stem cells and their power. The eye has also benefited from stem cell therapies. Certain forms of blindness, such as those caused by damage to the cornea, can be treated with stem cell transplants. In these therapies, limbal stem cells located at the edge of the cornea are used to regenerate damaged tissue, restoring clarity and vision. Patients who once lived in darkness have regained sight, not through mechanical devices or artificial implants, but through the quiet renewal of their own tissue. This work shows how even delicate organs can respond to the restorative presence of stem cells. These established therapies reveal the practical value of stem cells in medicine, but they are only the beginning. Researchers are exploring a wide range of new applications. In the field of cardiology, for example, scientists are investigating whether stem cells can repair heart muscle damaged by heart attacks. The heart, unlike the skin or blood, does not naturally regenerate well. When heart muscle cells die, they are replaced with scar tissue that cannot contract. The hope is that stem cells, guided to become cardiac muscle cells, could be introduced into the heart, where they might integrate into the tissue and restore lost function. Early studies show promise, though challenges remain. In neurology, stem cell research is being directed toward diseases such as Parkinson's, Alzheimer's, and spinal cord injuries. In Parkinson's disease, the loss of dopamine-producing neurons leads to tremors, stiffness, and difficulty with movement. Scientists are exploring whether stem cells can be coaxed into becoming these neurons and transplanted into the brain to restore function. For spinal cord injuries, where communication between the brain and body is disrupted, stem cells may one day provide new pathways by replacing damaged nerve cells. Though still in early stages, these possibilities suggest that conditions long considered incurable may eventually be addressed through the healing role of stem cells. Diabetes is another condition where stem cell therapy holds potential. In type 1 diabetes, the immune system destroys the insulin-producing cells of the pancreas. Without insulin, blood sugar cannot be controlled, and patients require lifelong treatment. Researchers are working to guide stem cells into becoming insulin-producing beta cells, with the hope that these cells could be transplanted into patients to restore natural insulin production. Success in this area could transform the lives of millions, offering a cure rather than a lifelong management strategy. Even in the field of organ transplantation, stem cells may play a role in the future. One of the greatest challenges of transplantation is the risk of rejection, where the immune system attacks the donor organ as foreign. Because stem cells can be taken from a patient's own body and reprogrammed into many cell types, it may one day be possible to grow replacement organs that are genetically matched to the recipient. Such organs would not be rejected, reducing the need for lifelong medications and greatly expanding the possibilities for transplantation. The use of stem cells in therapy also raises questions of safety and control. Stem cells must be guided carefully to prevent uncontrolled growth, which could lead to tumors. Researchers are developing methods to ensure that stem cells differentiate into the desired cell types before being used in patients. They are also studying the signals that control stem cell behavior, learning how to create environments that encourage healthy renewal while avoiding risks. These challenges remind us that the power of stem cells must be approached with patience and precision, balancing hope with caution. Even with these challenges, the presence of stem cells in medical therapy offers a profound sense of possibility. They are not simply tools of the future. They are already shaping treatments in the present. They remind us that healing is not always about adding something foreign to the body, but about guiding and supporting the natural processes that are already there. Stem cell therapies work by restoring what is missing, by amplifying what the body already knows how to do, by using renewal to replace loss. It is calming to imagine the role of stem cells in medicine as a gentle partnership between science and nature. Science provides the guidance, the environment, the tools to shape stem cells toward particular ends. Nature provides the cells themselves, with their innate ability to divide, renew, and differentiate. Together, they create therapies that are not imposed upon the body, but drawn from its own potential. The result is medicine that feels less like intervention and more like restoration, less like alteration and more like support. In the end, stem cells in medical therapy represent one of the most hopeful intersections of biology and healing. They remind us that the body carries within it the power to restore itself, and that science, by understanding and guiding this power, can transform lives. From the marrow that restores blood to the skin that heals burns, from the eyes that regain vision to the hearts and brains that may one day be renewed, stem cells are rewriting what is possible in medicine. They are a quiet revolution, one that works not with force but with patience, not with replacement but with renewal. To reflect on them is to see medicine not only as treatment but as partnership, a harmony between human knowledge and the quiet wisdom of life itself. 13. Experimental frontiers. The story of stem cells is not confined to what is already possible today. Much of the excitement surrounding these remarkable cells lies in what may yet come, in the therapies and treatments that are still being explored. These experimental frontiers of stem cell research are not simply distant dreams. They are areas of careful study, where scientists are testing, refining, and learning how to guide stem cells into roles that could transform medicine. To reflect on these frontiers is to look into the horizon of healing, where the body's own capacity for renewal is combined with human understanding, to tackle conditions that have long resisted cure. One of the most active areas of experimental research is in the treatment of neurodegenerative diseases, such as Parkinson's and Alzheimer's. Parkinson's disease arises when dopamine-producing neurons in a part of the brain called the substantia nigra are gradually lost. Without dopamine, the brain cannot coordinate movement smoothly, leading to tremors, stiffness, and difficulty with balance. Current treatments can ease symptoms, but they do not restore the lost neurons. Stem cells, however, offer a possibility. By guiding stem cells to become dopamine-producing neurons, scientists hope to transplant them into the brain, where they might take root, integrate with existing networks, and restore function. Early studies in animals and small trials in humans suggest this approach may one day provide real relief, offering not just symptom management but restoration. Alzheimer's disease, with its gradual erosion of memory and identity, presents an even greater challenge. The causes are complex, involving the accumulation of proteins, inflammation, and the loss of neurons across many regions of the brain. Stem cell research in Alzheimer's focuses not only on replacing lost neurons but also on understanding the disease itself. By creating induced pluripotent stem cells from patients with Alzheimer's, scientists can study how the disease develops at the cellular level, observing changes in neurons grown in the laboratory. This approach may lead to new therapies, guided by insights into the earliest stages of the disease. Though the cure remains distant, stem cells provide a way to peer into the mystery, to see what is otherwise hidden. Spinal cord injuries are another frontier where stem cells bring hope. When the spinal cord is damaged, communication between the brain and the body is disrupted. Paralysis may follow, leaving individuals unable to move or feel below the site of injury. For a long time, this damage was thought to be permanent, as nerve cells do not easily regenerate. Yet stem cell research has begun to challenge this assumption. By transplanting neural stem cells or progenitor cells into damaged spinal cords, researchers have seen some evidence of new connections forming, of partial recovery of movement and sensation. The path is still long, and the challenges are immense, but each step forward brings the possibility that paralysis may not always be a final condition. The heart is also at the center of stem cell research. When a heart attack occurs, blood flow is blocked, and muscle cells die. The body responds by replacing the dead tissue with scar tissue, which provides structure but not contraction. The heart weakens, and the risk of failure grows. Researchers are exploring whether stem cells can repair this damage, producing new heart muscle cells that beat in rhythm with the rest of the heart. Trials using bone marrow stem cells and cardiac progenitor cells have shown mixed results. But progress continues. The vision is clear: a therapy that allows the heart to heal itself, reducing the burden of one of the world's most common causes of death. Diabetes, particularly type 1 diabetes, is another condition at the frontier of stem cell research. In type 1 diabetes, the immune system destroys the beta cells of the pancreas, which are responsible for producing insulin. Without insulin, blood sugar cannot be controlled, and patients rely on lifelong injections or devices. Stem cell research seeks to replace these lost beta cells. By guiding stem cells to become insulin-producing cells, scientists hope to transplant them into patients, restoring natural regulation of blood sugar. Progress has been made, with experimental therapies already showing that such cells can survive and function in humans. The challenge now lies in protecting them from immune attack, ensuring that they endure in the body long enough to make a lasting difference. The eyes, delicate organs that give us vision, are also a focus of experimental therapies. Conditions such as macular degeneration, which cause the gradual loss of central vision, have been difficult to treat. Stem cells offer the possibility of replacing damaged retinal cells, restoring sight where it has been lost. Early clinical trials have shown encouraging results, with patients experiencing improvements in vision after receiving stem cell-derived retinal cells. Though these treatments are still experimental, they carry the promise of turning blindness into sight, not through machines or implants, but through the natural renewal of living cells. Another experimental frontier lies in the study of organ regeneration. Scientists are exploring whether stem cells can be used to grow entire organs in the laboratory, such as kidneys, livers, or even hearts. These efforts are still in their infancy, but progress has been made in creating organoids, miniature versions of organs grown from stem cells. These organoids cannot yet replace full organs, but they allow researchers to study development and disease in ways that were once impossible. They may also serve as testing grounds for drugs, reducing the need for animal experiments and offering insights into human biology. One day, the vision is that organs grown from a patient's own cells could be transplanted, eliminating the problems of rejection and scarcity. Cancer, too, has drawn attention in stem cell research. Many cancers are now thought to involve cancer stem cells, cells that drive the growth and recurrence of tumors. By studying these cells, scientists hope to develop treatments that target the root of cancer rather than just its bulk. Stem cells are also being studied as vehicles for delivering therapies, engineered to carry drugs or signals directly into tumors. These approaches are still experimental, but they highlight the flexibility of stem cell science, its ability to adapt to challenges across many fields of medicine. The experimental frontiers of stem cells are vast and varied, but they are united by a common theme: the idea that renewal can replace loss, that cells can be guided to restore what disease or injury has taken away. Each step forward is cautious, tested carefully for safety and effectiveness. Progress is sometimes slow, but the potential is immense. These therapies are not yet routine, but they are no longer distant fantasies. They are real studies, real trials, real hopes in the process of becoming reality. It is soothing to think of stem cells at the frontier of medicine as pioneers of healing. They move into areas where no other treatments have succeeded, exploring possibilities where hope was once absent. They represent not only the science of renewal but also the imagination of healing, the belief that what seems irreversible might one day be restored. The frontier is not a single place, but many. In the brain, the heart, the pancreas, the eyes, the spine. Each frontier is a reminder that the story of stem cells is still unfolding, that their potential is as open as their nature, and that renewal is not only present but always ahead, waiting to be discovered. 14. Stem cells and cloning. Cloning is a word that often stirs images of science fiction, of entire organisms replicated in laboratories, of identical beings created by design. Yet within the calm rhythm of biology, cloning has a more subtle and grounded meaning. At its heart, cloning is about creating a copy, whether of a gene, a cell, or, in some cases, an entire organism. Stem cells and cloning intersect in ways that illuminate not only the potential of life to renew itself, but also the ability of science to guide and replicate that potential. This intersection is sometimes called therapeutic cloning, and it stands as one of the most fascinating frontiers of biology, a place where the concept of renewal and the idea of replication come together. To understand how stem cells and cloning connect, it helps to recall that every cell in the human body carries the same DNA, the same complete set of instructions for building an organism. A skin cell, a muscle cell, and a neuron all contain the same genetic code. What makes them different is not the DNA itself, but which parts of it are active, which chapters of the book are being read. The realization that all of an organism's information is stored in every cell opened the door to the idea of cloning. If one could reset a cell, erasing its specialized identity, then perhaps it could be guided to recreate the entire organism, or more subtly, provide new stem cells for healing. This resetting is the essence of a process known as somatic cell nuclear transfer. In this technique, the nucleus of a specialized adult cell is removed and placed into an egg cell that has had its own nucleus removed. The egg cell, with its remarkable ability to direct development, reprograms the adult nucleus, wiping away its specialized identity and returning it to a state of openness. The result is a cell that can begin dividing like an embryonic cell, producing a cluster of pluripotent stem cells. From here, the cells can be guided into becoming any type of tissue. This approach is called therapeutic cloning when it is used not to create an entire organism, but to create stem cells for research and treatment. The advantage of therapeutic cloning lies in the fact that the stem cells produced are genetically identical to the donor of the original nucleus. This means they can be used for therapies without the risk of immune rejection, since the body recognizes them as its own. For patients with genetic diseases, therapeutic cloning could provide a way to create healthy tissues matched perfectly to their DNA, offering personalized healing drawn from their very own cells. One can imagine, for example, a patient with damaged heart tissue. Through therapeutic cloning, scientists could create stem cells genetically identical to the patient, guide them into becoming heart muscle cells, and use them to repair the damaged organ. The same could be done for neurons lost to Parkinson's disease or insulin-producing cells destroyed in type 1 diabetes. The promise is that cloning, in this therapeutic sense, allows stem cells to be tailored perfectly to each individual, avoiding the complications of rejection and providing truly personalized medicine. Cloning and stem cells also intersect in the study of disease. By creating stem cells from patients with specific genetic disorders, researchers can grow tissues in the laboratory that mirror the disease's effects. These tissues can then be studied to understand how the condition develops and how it might be treated. For example, scientists have used cloned stem cells to study neurological diseases, growing neurons that carry the genetic mutations responsible. Observing these neurons in controlled settings provides insights that could never be gained directly from patients, and it allows therapies to be tested in precise and personal ways. Of course, cloning also carries the idea of reproductive cloning, where the goal is to create an entire organism. This is the form of cloning that captured the world's attention with the birth of Dolly, the sheep, in 1996. The first mammal cloned from an adult cell. Dolly's creation showed that it was indeed possible to take the nucleus of a specialized cell, reset it, and guide it to form a complete individual. While fascinating, reproductive cloning raises profound ethical questions, particularly when considered in the context of humans. Most scientific efforts today focus not on creating entire organisms but on therapeutic cloning, where the goal is healing rather than replication. It is calming to reflect on therapeutic cloning not as an act of duplicating life but as an act of supporting it. It is not about creating identical beings but about restoring balance within a single body. It is about using the natural ability of cells to be reprogrammed, guided, and renewed to replace what has been lost. The focus is on healing tissues, on repairing organs, on giving patients back what disease or injury has taken away. In this sense, therapeutic cloning is not a step toward science fiction, but a continuation of nature's own capacity for renewal, carefully directed by human knowledge. The connection between stem cells and cloning also deepens our understanding of life's plasticity. The fact that a specialized cell, such as a skin cell, can be returned to an embryonic state through reprogramming shows that identity within the cell is not as rigid as once thought. Life carries within it a flexibility that can be revealed under the right conditions. This truth echoes what has been learned from induced pluripotent stem cells, where reprogramming is achieved by introducing specific genes rather than transferring nuclei. Both approaches point to the same lesson. Cells can change, and potential can be restored. In thinking about cloning, it is easy to become caught up in the technical details, the procedures, and the debates. But it is also worth pausing to consider the deeper imagery. One might picture a stem cell as a spark of possibility, and cloning as the act of rekindling that spark within a cell that had long seemed fixed. It is a reminder that within life lies the power not only to persist but also to return, to be renewed, to begin again, from what once seemed finished. The ethical considerations surrounding cloning remind us that science always moves hand in hand with responsibility. Questions about the origins of embryos, the purposes of cloning, and the boundaries of its use are all part of the conversation. These questions ensure that progress is thoughtful, that discovery is tempered with reflection. In the balance between promise and principle, Stem cells and cloning find their place as both tools of healing and subjects of care. In the end, stem cells and cloning together tell a story of renewal through replication. They reveal that cells, even when specialized, carry the full potential of life within them, waiting to be unlocked. They show that the body's own materials can be used to heal itself, that identity can be reset, that possibility is never entirely lost. Therapeutic cloning, in particular, is a gentle reminder that healing can be drawn from within, that the answers to disease and injury may lie in the very cells of those affected. To reflect on this is to see cloning not as a dramatic leap, but as a quiet unfolding of life's own flexibility, guided by the steady hands of science toward the service of renewal. 15. Regenerative medicine and the future. Regeneration is a word that carries with it a sense of renewal, of life returning where it had been diminished, of tissues and organs restored after loss. In the natural world, regeneration takes many forms. Some animals, like salamanders, can regrow entire limbs, while others can replace parts of organs or tissues when damaged. Humans do not share this same dramatic capacity, yet within us lies a quiet version of regeneration, carried out by stem cells. It is this hidden power that has given rise to the field of regenerative medicine, a branch of science and therapy devoted to guiding and amplifying the body's natural ability to heal. Where once medicine focused on treating symptoms or slowing decline, regenerative medicine looks toward restoration, aiming to replace what has been lost, not simply to ease its absence. At the core of regenerative medicine are stem cells, the versatile builders of new tissue. Their ability to self-renew and to differentiate into many types of specialized cells makes them uniquely suited to the task of repair. In the vision of regenerative medicine, stem cells are not only the body's silent workers, but also tools that can be directed, transplanted, and guided into creating new tissues and even new organs. The dream is to move beyond managing disease and instead restore full health, giving patients back the functions they have lost. One of the most promising areas of regenerative medicine lies in the treatment of heart disease. When a heart attack occurs, blood supply to part of the heart muscle is blocked, and cells die. The body responds by forming scar tissue, which cannot contract or conduct electrical signals. The result is a weakened heart, less able to pump blood. Conventional treatments can reduce strain or prevent further damage, but they cannot replace the lost muscle. Regenerative medicine aims to change this by introducing stem cells into the heart, or by guiding stem cells to become cardiac muscle in the laboratory, and then transplanting them. Scientists hope to repair damaged hearts. Imagine a future in which heart attacks no longer lead to permanent decline, but to healing, as new muscle grows and integrates with the old. In the nervous system, regenerative medicine seeks to restore connections lost through injury or disease. The spinal cord, once injured, has little ability to heal itself. Paralysis often follows, the communication between brain and body severed. Yet stem cell research offers hope that new neurons and glial cells can be introduced, bridging damaged regions and re-establishing pathways. Clinical trials are already testing whether neural stem cells can improve function in patients with spinal cord injuries. Though the progress is careful and gradual, each advance moves closer to a vision where paralysis may not always be permanent. The eyes also stand at the forefront of regenerative efforts. Diseases such as macular degeneration cause the slow loss of retinal cells, leading to blindness. Stem cells offer the possibility of replacing these cells, restoring sight not through artificial lenses or devices, but through living tissue renewed. Patients who once faced inevitable loss of vision may one day receive stem cell-derived retinal cells, allowing them to see again. Already, early clinical trials have shown that such approaches are possible, that sight can be improved by restoring the natural architecture of the eye. Regenerative medicine also looks toward the liver, an organ already known for its remarkable capacity to repair itself. The liver can regenerate portions of its tissue after damage. Yet in chronic disease or severe injury, this capacity is overwhelmed. Stem cells may provide a way to support or extend the liver's natural healing, replacing damaged tissue and restoring function. Similar efforts are being studied in the kidneys, lungs, and pancreas, organs that do not naturally regenerate well but may be renewed through stem cell therapy. Perhaps the most ambitious vision of regenerative medicine is the idea of growing entire organs in the laboratory. With the shortage of donor organs a constant challenge, scientists are exploring ways to use stem cells to create new organs from scratch. Already, small organoids, miniature versions of organs, have been grown from stem cells. These organoids do not yet replace full organs, but they serve as models for studying disease and testing drugs. One day, the hope is that full-sized, functional organs could be grown for transplantation, matched genetically to the patient to prevent rejection. This vision is bold, but it is rooted in the quiet truth that stem cells carry the instructions to create tissues, if only they can be guided in the right way. The techniques of regenerative medicine are varied. Sometimes stem cells are transplanted directly, introduced into damaged tissue where they can integrate and repair. Other times they are grown into specific cell types in the laboratory, then transplanted into patients. In some approaches, scientists use scaffolds, structures that provide a framework on which stem cells can grow into organized tissues. In others, they use signals and molecules to encourage stem cells already within the body to awaken and repair. Each technique is part of a growing toolbox, a collection of methods that draw on the natural flexibility of stem cells to restore health. As with all frontiers of medicine, regenerative therapies bring challenges as well as promise. One challenge is ensuring that stem cells behave as intended, differentiating into the right types of cells without uncontrolled growth. Another is ensuring that transplanted cells survive, integrate, and function properly within the body. Immune rejection remains a risk, though approaches using a patient's own cells, reprogrammed into pluripotent states, may reduce this. Despite these challenges, progress continues steadily, guided by caution and care. What makes regenerative medicine so compelling is not only its technical promise but its philosophical shift. It represents a move away from treating decline as inevitable and toward seeing restoration as possible. It views the body not as something that can only be patched and supported, but as something that can be renewed. It suggests that disease and injury, while real and challenging, may not always have the final word. Instead, the final word may belong to renewal, drawn from the body's own hidden reserves. It is soothing to imagine a future shaped by regenerative medicine. A patient who suffers a heart attack may recover not with permanent weakness, but with restored strength. Someone who loses vision may see again, not through a machine, but through their own renewed cells. A person facing paralysis may regain movement as new neurons restore connections. Even those waiting for organs may find hope in tissues grown from their own stem cells. Each of these possibilities is an extension of what stem cells already do quietly every day: repair, renew, and replace. In reflecting on regenerative medicine, one sees not only a field of science, but a metaphor for life itself. It reminds us that loss does not always mean permanence, that decline is not always final, and that renewal is built into the very fabric of our biology. Stem cells, with their silent persistence, embody this truth. Regenerative medicine is the art of guiding them, of amplifying what they already know, of using their potential to restore what has been lost. In the end, regenerative medicine and stem cells together form a vision of the future where healing is not about managing decline but about rediscovering wholeness. It is a future in which medicine is not only reactive but restorative, not only supportive but transformative. To reflect on this future is to rest with hope, a calm assurance that within the smallest parts of ourselves lies the power to heal, and that science is learning how to bring this power gently into the service of life. 16. And philosophical reflections. Whenever science ventures into the delicate realms of life's beginnings and the essence of renewal, it naturally invites questions that extend beyond biology. Stem cells, with their extraordinary ability to become many things, are not only scientific wonders but also sources of deep ethical and philosophical reflection. They touch on the meaning of life, the boundaries of medicine, the nature of identity, and the responsibilities that come with knowledge. To understand stem cells fully is not only to examine their functions but also to consider the questions they raise about what it means to heal, to begin, and to respect the sanctity of living matter. One of the earliest and most enduring ethical debates about stem cells has centered on embryonic stem cells. These cells, drawn from embryos in the earliest stages of development, are unique because they are pluripotent, capable of becoming every tissue in the body. Their promise is immense, but their source has raised difficult questions. For some, the embryo represents the earliest form of human life, deserving protection and dignity from the moment of conception. For others, the use of embryos in research is justified when it can lead to treatments that save lives, reduce suffering, and restore health to countless individuals. Between these views lies a wide spectrum of perspectives, reflecting different cultural, religious, and philosophical beliefs about when life begins and what responsibilities accompany it. The debate over embryonic stem cells highlights a broader theme: the tension between potential and responsibility. Stem cells carry the potential to cure diseases, to repair organs, to transform medicine, yet the responsibility lies in how they are obtained, how they are used, and what boundaries are respected. Science often advances in ways that require careful reflection, balancing the pursuit of knowledge with the values that guide human society. The history of stem cell research reflects this balance, as ethical concerns have led to regulations, alternative methods, and the development of new techniques, such as induced pluripotent stem cells, which allow researchers to avoid the use of embryos altogether. Induced pluripotent stem cells, created by reprogramming adult cells into a pluripotent state, offer a way to sidestep some of the ethical concerns surrounding embryonic stem cells. They demonstrate that science can adapt, finding solutions that honor both the pursuit of healing and the respect for ethical boundaries. Yet even with these alternatives, questions remain. How should such powerful technologies be used? Who should have access to them? What safeguards are needed to ensure that they are applied with fairness and care? These are not questions of science alone, but of philosophy, justice, and the shared values of humanity. Another area of reflection involves the boundaries of medicine itself. Stem cells offer the possibility of not only treating disease, but also enhancing human abilities. If stem cells can repair tissues, could they also be used to make them stronger? If they can restore aging organs, could they also slow the process of aging itself? These questions touch on the distinction between therapy and enhancement, between healing what is broken and altering what is already whole. Some see the extension of stem cell use into enhancement as a natural continuation of medicine's role in improving life. Others see it as a step too far, one that risks altering the balance of nature or creating inequalities between those who can access such treatments and those who cannot. The philosophical reflections extend even deeper to the question of identity itself. Stem cells remind us that every cell in the body carries the same genetic code, that a skin cell could, in theory, be reprogrammed into a neuron, that the boundaries between one type of cell and another are not absolute. This plasticity raises questions about what makes something Whatted Island. If a cell can change its identity, then identity in biology is not fixed, but fluid, shaped by environment, signals, and choices. This flexibility mirrors broader reflections on identity in human life, suggesting that change, renewal, and transformation are not exceptions but natural truths of existence. Stem cells also challenge us to reflect on the relationship between the individual and the collective. Therapeutic cloning, for example, offers the possibility of creating stem cells that are genetically identical to an individual, tailored perfectly to their body. This speaks to the uniqueness of each person, the intimate connection between their cells and their identity. Yet at the same time, stem cells remind us of shared humanity, the principles that govern their renewal, the ways they repair and sustain, are the same in all people. In this sense, stem cells represent both individuality and universality, showing that we are at once unique and profoundly connected. There is also the matter of justice, of how the benefits of stem cell research and therapy are distributed. These therapies are often complex, expensive, and tied to advanced medical systems. Philosophically, one must ask, who has the right to renewal? Should the ability to regenerate tissues or cure diseases be limited to those with resources? Or should it be a shared gift of humanity? The answers to such questions lie not only in laboratories but in societies, in the decisions we make about equity, access, and the common good. The presence of ethical reflection does not diminish the promise of stem cells. Rather, it deepens it, reminding us that healing is not only a biological process, but also a moral one. The power to restore tissues, to repair hearts, to renew sight, is not only technical but also human. It requires care, humility, and a recognition of the responsibilities that accompany such power. The questions are not obstacles but companions to discovery, guiding progress with thoughtfulness and restraint. Philosophically, stem cells also invite reflection on the nature of life itself. They embody renewal, showing that life is not static but dynamic, not fixed but fluid. They remind us that decline is not absolute, that within every organism lies the power to regenerate. This truth resonates beyond biology, echoing in the human experience. Renewal is possible not only in cells but in lives, in communities, in societies. Stem cells, in their quiet persistence, become symbols of resilience, teaching that restoration is always possible, even when decline seems inevitable. It is soothing to imagine stem cells not only as scientific tools but as reminders of balance. They hold within them both immense potential and the need for restraint, both the promise of healing and the call for responsibility. They ask us to move forward with care, to honor both the power of science and the values of life. They suggest that progress is not only about what can be done, but about what should be done and how it should be shared. In the end, the ethical and philosophical reflections surrounding stem cells are not separate from their science, but intertwined with it. They shape the way research is conducted, the way therapies are applied, and the way discoveries are integrated into society. They remind us that renewal is not only a matter of biology, but also of choice, principle, and shared humanity. To reflect on them is to see that the promise of stem cells lies not only in their ability to heal bodies, but also in their ability to invite deeper thought about what it means to live, to begin, and to honor life itself. 17. Stem cells in everyday life. Though the research and headlines surrounding stem cells often focus on dramatic medical applications and the promise of futuristic therapies, their quiet presence in everyday life is no less remarkable. Long before scientists began to study them, stem cells were already at work, woven into the rhythm of living, maintaining the body in silence. They are not visitors that appear only in times of illness or injury, but constant companions, active from the earliest moment of existence and persisting until life's final day. In every hour, in every tissue, they contribute to the ordinary processes that allow the body to endure. Without them, even the most basic functions of daily survival would collapse. The blood provides one of the clearest examples of this ongoing presence. Every day, billions of blood cells are created and destroyed. Red blood cells, which carry oxygen, live for only about 4 months before they are broken down and replaced. White blood cells, which fight infection, live for much shorter spans, sometimes just a few days. Platelets, fragments that help the blood clot, survive only about a week. If this constant renewal were to pause even for a short time, circulation would falter, oxygen would not reach the tissues, infections would overwhelm the body, and wounds would not close. The reason this does not happen is because hematopoietic stem cells in the bone marrow never stop dividing. They provide an unbroken supply of blood cells, replenishing the river of life without interruption. The skin is another vivid example. It is the body's largest organ, and its surface is in constant contact with the outside world. Sunlight, friction, dust, microbes, and countless other factors wear down its protective barrier. The outermost cells, flattened and hardened, live only briefly before they flake away. Yet the skin never grows thin or bare because stem cells in the basal layer of the epidermis quietly divide, sending upward new cells to take the place of those that are lost. This cycle of shedding and renewal is so steady that within a month, most of the surface of the skin has been replaced entirely. We rarely notice this renewal, but it is happening continually, an unseen act of preservation. The lining of the gut provides perhaps the most dramatic example of stem cells at work in everyday life. The intestinal lining is exposed constantly to the passage of food, liquid, and microbes. It is a place of absorption and defense, and it is subject to continuous wear. To withstand this challenge, intestinal stem cells generate new cells at an astonishing pace. Within just a few days, the entire lining of the intestine is renewed. Old cells are shed, and fresh ones take their place, maintaining the delicate balance that allows digestion and protection to occur at once. Without this renewal, the gut would quickly erode, leaving the body unable to process nourishment or defend itself. Stem cells also contribute to the repair of muscles after even the smallest strains of daily life. When muscles are used in ordinary activities, walking, lifting, stretching, tiny fibers may sustain microtears. These are not injuries in the dramatic sense, but normal outcomes of use. Muscle stem cells, known as satellite cells, respond quietly to these small disruptions, fusing with fibers to repair and strengthen them. Over time, this process allows muscles to adapt, becoming stronger and more resilient with use. Without these stem cells, even the ordinary act of walking might gradually weaken the muscles beyond repair. Hair growth is another everyday rhythm sustained by stem cells. In the follicles beneath the skin, stem cells divide in cycles, producing the keratinocytes that form the shafts of hair. This cycle of growth, rest, and renewal continues quietly throughout life, reflecting the same principles of persistence and adaptation found in other tissues. The presence of stem cells ensures that hair continues to grow, even as individual strands fall away. Even in the mouth, stem cells are quietly at work. The lining of the oral cavity, like the skin and gut, is subject to constant friction and wear from eating, speaking, and chewing. Stem cells here renew the tissues with remarkable speed, maintaining a surface that is resilient and resistant to damage. Their presence allows the mouth to heal quickly from small cuts or abrasions, preserving one of the body's most important gateways. Though less visible, the brain too is supported by stem cells in everyday life. In regions such as the hippocampus, new neurons are generated from neural stem cells, contributing to memory, learning, and adaptation. The birth of new neurons may not be dramatic, but it provides a subtle flexibility that helps the brain respond to change and experience. Even in ordinary days, as we form new memories or learn new skills, neural stem cells play their quiet part, weaving renewal into the tapestry of thought. The immune system is another arena where stem cells work constantly. Infections, even minor ones, consume vast numbers of white blood cells. Without a steady supply of new ones, the defenses of the body would quickly collapse. Stem cells ensure that there is always a reserve, always fresh soldiers ready to defend against invaders. Even in times of health, they maintain this supply, preparing the body for challenges that may come. The ordinary resilience we feel in daily life, the ability to withstand colds, to heal from small illnesses, to remain well, is made possible by this quiet renewal. The ordinary work of stem cells extends to countless other tissues. The cornea of the eye, where they renew the transparent surface that allows vision. The liver, where they help support one of the body's most versatile organs. And even the lining of the respiratory tract, where they replace cells worn down by the passage of air. In each case, the pattern is the same. Cells are lost, and stem cells replace them. Tissues wear, and stem cells restore them. The body continues not through permanence but through renewal, not through resistance to change but through constant adaptation. To reflect on stem cells in everyday life is to recognize that survival is not a static achievement but a dynamic process. The body is never the same from one moment to the next. Cells die, and new ones are born. Tissues erode, and they are rebuilt. The appearance of stability is created by constant change, by the hidden rhythm of stem cells at work. This truth can be deeply calming, for it shows that resilience is not about avoiding loss, but about responding to it. Not about resisting time, but about adapting to it. It is soothing to imagine stem cells as quiet caretakers, scattered throughout the body like lanterns in the night. Some glow brightly, always active, as in the gut. Others glow softly, waiting for a signal, as in the muscles or the brain. Together, they form a constellation of renewal, a network of persistence that sustains life, moment by moment. We rarely notice their presence, yet they are always there, shaping the continuity of life without pause. In the end, stem cells in everyday life remind us that health itself is not the absence of change. They show us that the body is not a fixed structure but a living process, always remaking itself, always adapting to the challenges of the world. To reflect on them is to see that life is not fragile because it changes, but resilient because it renews. The quiet work of stem cells is the foundation of that resilience, the reason that the body endures not just in extraordinary moments, but in the ordinary passage of days. 18. Stem cells as a bridge. Stem cells occupy a unique position in the story of life. They are not just participants in one part of biology, but a bridge across many of its landscapes. They connect the beginnings of development to the persistence of adulthood, the renewal of tissues to the possibilities of medicine, the mysteries of evolution to the daily rhythms of survival. To reflect on stem cells is to see how they stand at the intersections of biology, uniting areas that might otherwise seem separate. They are at once the architects of growth, the guardians of health, and the keys to understanding how life adapts and endures. From the perspective of development, stem cells are the starting point. Every human life begins as a single fertilized cell, which divides into many, and from these arise pluripotent stem cells in the early embryo. These embryonic stem cells form the bridge from a single cell to a complex organism. Without them, there could be no differentiation, no tissues, no organs. They hold the capacity to become all parts of the body, linking the potential of beginnings to the structure of life as it takes shape. In this sense, they connect the simplicity of origins with the complexity of form. But the bridge does not end with development. A different population of stem cells carries us into adulthood, sustaining the body once it has been built. Adult stem cells, though more limited in their potential, continue the work of preservation and repair. They link growth to maintenance, ensuring that what was formed in the beginning does not fall apart. Hematopoietic stem cells, for example, connect embryonic development to lifelong survival by continually renewing the blood. Skin stem cells connect the fragile outer layer of the embryo to the durable barrier of adult life. Intestinal stem cells connect the unfolding of the gut in the fetus to the ongoing work of digestion in maturity. Through these examples, Stem cells show how life builds a bridge, not only between moments but across the whole arc of existence. Stem cells also serve as a bridge between injury and recovery. Damage is inevitable, whether through accident, disease, or the natural wear of time. Without repair, life would soon collapse under the weight of injury. Yet stem cells provide a link between loss and renewal. They step in to replace what is gone, to rebuild what is broken, to refresh what is worn. When a bone breaks, stem cells connect the fracture to its healing. When skin is cut, they link the wound to its closure. When muscles are strained, they bridge the gap between weakness and strength. Their role is not dramatic but essential, transforming harm into healing. In the laboratory, stem cells form another kind of bridge between science and medicine. For researchers, they are windows into development, allowing the earliest processes of life to be observed and understood. For doctors, they are tools of therapy, offering ways to restore health. In this way, stem cells connect the abstract pursuit of knowledge with the practical goal of healing. The same cells that teach scientists about embryonic growth also offer clinicians the possibility of treating leukemia, repairing burns, or restoring sight. They unite curiosity and compassion, discovery and care, within a single field of study. Stem cells also bridge the present with the future. They represent not only what medicine can do today, but what it may be able to do tomorrow. Bone marrow transplants, skin grafts, and corneal repairs are established therapies already saving lives. Yet the same principles are being extended toward new horizons, regenerating hearts, repairing spinal cords, restoring neurons, and even growing new organs. The work is ongoing. The challenge is real, but the bridge is already being built, linking the treatments of today to the possibilities of tomorrow. Stem cells carry us across this passage, step by step, from established practices to dreams in the making. From an evolutionary perspective, stem cells also play a bridging role. They represent the continuity of life's strategy for resilience. Across species, from simple organisms to complex mammals, stem-like cells are present, providing the capacity to regenerate. In some animals, this capacity is expansive. Salamanders can regrow limbs, flatworms can regenerate entire bodies. In humans, the capacity is more modest, limited to certain tissues, yet the principle is the same. Stem cells connect us to the broader story of life, showing how the ability to renew is not unique but shared, a strategy refined through millions of years of evolution. They remind us that resilience is a common thread, a bridge woven through the tapestry of all living things. On a philosophical level, stem cells bridge the tangible with the symbolic. They are real, measurable entities, studied under microscopes, guided in laboratories. Yet they also carry deeper meaning. They embody possibility, resilience, and renewal. They remind us that identity is not fixed, that potential persists, that healing is possible even after loss. They link science with metaphor, biology with philosophy, the material with the meaningful. To consider stem cells is not only to learn about cells, but also to reflect on the nature of life itself. In everyday life, too, stem cells form bridges between the unseen and the experienced. We do not notice them directly. We do not feel their divisions or perceive their renewal. Yet we experience the results, the skin that heals, the blood that flows, the muscles that recover, the memories that form. Stem cells bridge the hidden activity of the body with the visible experience of living. They connect silence with survival, invisibility with resilience. It is soothing to imagine these bridges as threads woven through the fabric of existence. Some are large and obvious, like the bridge from embryo to adult. Others are small and constant, like the bridge from injury to healing. Together, they create a network that sustains life, connecting one moment to the next, one process to another, one possibility to its fulfillment. Stem cells are not isolated players but connectors, linking the many dimensions of biology into a single, continuous flow. In the end, stem cells as a bridge remind us that life is not fragmented but whole. Development, maintenance, healing, medicine, and evolution are not separate stories but parts of one story. Connected by the quiet presence of these remarkable cells, they carry us from potential to reality, from damage to repair, from knowledge to application. They are the threads that hold the tapestry together, the arches that span the gaps, the links that make continuity possible. To reflect on them is to see that life itself is a bridge, carried forward by renewal, always connecting what was to what is, and what is to what will be. 19. Stem cells as resilience and renewal. The story of stem cells is, at its heart, a story of resilience. They are the quiet embodiment of the body's ability to endure, to adapt, and to begin again. From the very first moments of existence until the final chapters of life, stem cells persist as guardians of renewal. They are present in growth, in healing, and in the quiet turnover of everyday life. They remind us that resilience is not about avoiding change or preventing loss, but about meeting those realities with the power to restore, to refresh, and to renew. In their silent persistence, stem cells illustrate the essence of what it means for life to continue. From the beginning, resilience is written into the biology of stem cells. In the early embryo, pluripotent stem cells hold the ability to become anything. They form the foundation for the entire body, building complex tissues and organs from a state of openness. This is resilience in its purest form, the ability to adapt, to create, to respond to the needs of development with infinite flexibility. Without it, the embryo could not survive, could not grow into the intricate form of a human being. The fact that all tissues arise from such a foundation shows that renewal is not an afterthought in biology, but a starting principle, woven into life's very beginning. As life moves into growth and maturity, stem cells shift their role from creation to preservation. Adult stem cells, though more limited, continue to provide renewal across the years. In the bone marrow, hematopoietic stem cells ensure that the blood is never exhausted. In the skin, stem cells replenish the surface, maintaining a protective shield. In the intestines, they replace the lining at astonishing speed, preventing wear from overwhelming function. These processes are not dramatic but constant, ensuring that tissues endure despite the losses they sustain. Resilience here takes the form of quiet repetition, of cycles that never break. It shows that survival is not the absence of damage, but the ability to recover from it endlessly. Resilience also shines most clearly in times of injury. A broken bone is not the end of movement because stem cells rebuild it. A cut in the skin is not a permanent wound because stem cells close it. A muscle strained by effort is not lost because stem cells restore its fibers. Even when the body faces illness, such as infections that deplete immune cells, stem cells ensure that replacements are made, that the army of defense rises again. Each of these acts of healing is a reminder that resilience is not about never being harmed, but about always being able to return. As the body ages, the activity of stem cells begins to slow. Renewal does not cease, but it becomes less vigorous. Wounds take longer to heal. Tissues recover more slowly, and the balance between loss and replacement shifts. Yet, even in old age, stem cells remain, working quietly to sustain life. They are the reason the body continues to function for decades, the reason resilience does not vanish even when time has worn the body thin. In this way, stem cells embody renewal across the entire lifespan, carrying resilience from the first day of life to the last. Stem cells also symbolize resilience in the broader sense of medicine and science. They show that even when tissues are damaged beyond the body's natural ability to repair, renewal may still be possible through therapy. bone marrow transplants, burn treatments, corneal repairs, and experimental therapies for the heart, brain, and pancreas all build on the principle of stem cells. They extend the body's natural resilience, amplifying what stem cells already do and guiding them toward greater renewal. This reflects the resilience of human knowledge as well, the ability of science to adapt, to respond to challenges, and to find solutions where once there were none. There is a quiet comfort in knowing that resilience is not something extraordinary but something ordinary, present within the very cells of the body. Every day, as skin is shed, as blood cells die, as tissues wear, stem cells restore them. We rarely notice, but it is this constant renewal that allows us to continue. It suggests that resilience is not about dramatic recoveries alone, but about the small, repeated acts of persistence that sustain life. In this sense, stem cells are not only biological entities but also metaphors for how resilience works in all things. Through quiet endurance, steady renewal, and the refusal to stop adapting, it is calming to imagine stem cells as a network of small guardians, spread throughout the body like sparks of light. Some glow brightly in places of constant renewal, such as the gut and blood. Others glow softly, waiting for a time when they are needed, as in muscles or bone. Together, they form a constellation of resilience, a web of renewal that holds the body together through time. Even as individual cells die, the network remains, ensuring continuity. This imagery reflects not only the biology of stem cells but also the deeper truth of resilience itself. It is not about the permanence of individuals but about the persistence of the whole. Stem cells also reveal that resilience is not perfection. Renewal is not always complete. Scars form, tissues weaken with age, and some organs do not regenerate fully. Yet resilience does not require flawless restoration. It requires only the ability to adapt, to repair enough to continue, to endure, even when change has left its mark. Stem cells embody this kind of resilience. They do not always restore tissues exactly as they were, but they provide enough renewal for survival. In this way, they remind us that resilience is not about erasing the past but about living forward despite it. Philosophically, stem cells invite reflection on the nature of renewal in human life beyond biology. Just as stem cells restore tissues, people restore themselves in times of difficulty, communities rebuild after loss, and societies adapt to change. The biology of stem cells mirrors the rhythms of existence itself, showing that resilience is not an extraordinary trait of a few, but an ordinary truth of all life. Renewal is always possible, even after decline, even after damage. Stem cells are living proof of this principle, written into every body. In medicine, stem cells represent hope, but in philosophy, they represent reassurance. They tell us that life carries within it the capacity to restore itself, that decline is not always final, and that renewal is always present in some form. They suggest that resilience is not an effort imposed from outside, but a natural process within. To think of them is to rest with the comfort that renewal is always at work, silently and persistently, sustaining the body in ways we never see. In the end, stem cells as resilience and renewal remind us that life is not fragile because it changes, but strong because it renews. They are the hidden strength of biology, the reason wounds heal, the reason tissues endure, the reason life continues across decades. They are not dramatic but persistent, not loud but steady. They show that resilience is not about resisting time or harm, but about adapting to them, always finding ways to continue. To reflect on them is to reflect on the quiet strength that sustains all living things, the strength of renewal that never ceases, even in silence. 20. Quiet foundation. As we arrive at the final reflection on stem cells, It is fitting to step back and view them not only as scientific curiosities or tools of medicine, but as the quiet foundation of life itself. From the very first flicker of existence, when a single cell begins to divide, to the last moments of aging, when tissues still strive to repair themselves, stem cells are present. They do not call attention to themselves, nor do they announce their importance, but they are there all the same, weaving continuity into the fabric of being. They are not dramatic heroes of biology, but gentle sustainers, working in silence to make life possible. Every life begins with stem cells. In the earliest days after conception, embryonic stem cells hold within them the possibility of every tissue, every organ, every system. They are like a vast blank canvas upon which the story of the body will be painted. From them emerge the first outlines of form, the first divisions into layers, the first shaping of what will become heart, brain, skin, and bone. Without them, there could be no beginnings, no unfolding of structure from simplicity. They represent the openness of possibility at the dawn of existence, a reminder that life starts not with limitation, but with expansiveness. As life progresses, stem cells remain. They shift roles, from architects to caretakers, from builders to maintainers. Adult stem cells, though less versatile, continue the work of renewal. They replace worn blood cells, refresh the lining of the gut, regenerate skin, repair bone, and mend muscle. Their persistence allows the body to endure the passage of time. Each day, countless cells die, and countless new ones are born, creating an illusion of permanence built upon constant change. This unending rhythm of replacement is sustained by stem cells, the foundation beneath the everyday processes of life. When injury strikes, stem cells take on yet another role. They become healers, summoned by the signals of damage to repair what has been broken. A cut in the skin, a fracture in the bone, a strain in the muscle, all these call stem cells into action. They divide, differentiate, and restore, turning harm into recovery. Their work is rarely perfect. Scars form, tissues may not be identical to what was lost, yet the principle remains. Life does not simply collapse in the face of injury, but responds with renewal. Stem cells are the agents of that renewal, the foundation of resilience that allows bodies to recover again and again. In medicine, stem cells have expanded from their quiet natural roles into deliberate tools of healing. Bone marrow transplants, skin grafts, corneal repairs. All these therapies are possible because stem cells can be harnessed and guided. In experimental frontiers, they promise even more: the repair of hearts, the regeneration of neurons, the restoration of insulin production, the growth of new organs. Medicine has begun to align itself with the natural wisdom of stem cells, using their power not to impose something artificial, but to amplify what the body already knows. This union of biology and science is itself a reflection of the foundational role stem cells play, bridging nature's renewal with human intention. Philosophically, stem cells invite reflection on the nature of continuity. They show that life is not a fixed state, but a process, always in motion, always remaking itself. They demonstrate that permanence arises not from immobility, but from constant renewal. In this way, stem cells serve as metaphors as much as biological realities. They suggest that resilience in life comes not from resisting change, but from embracing it, not from holding tightly to the old, but from allowing the new to arise. Their quiet persistence mirrors the rhythms of human existence, where loss and renewal are constant companions. They also remind us of the fluidity of identity. A stem cell, unspecialized, can become many things. Its fate is not predetermined, but guided by signals, environment, and need. This flexibility reflects the truth that identity is not rigid but adaptive, shaped by context and possibility. Just as stem cells adapt to the needs of the body, so too do people adapt to the circumstances of life. In this sense, stem cells are not only foundations of biology, but quiet teachers of philosophy, illustrating principles of adaptability, openness, and renewal. The quiet foundation they provide is also universal. Though much of the discussion focuses on humans, stem cells are present in all forms of life. In simple organisms, they give rise to entire bodies from fragments. In complex mammals, they maintain tissues and enable healing. Across species, the principle is the same. Life endures because it can renew. Stem cells connect humanity to the broader web of life, showing that resilience is not ours alone, but a shared truth across the natural world. Even in aging, when the vigor of stem cells declines, their presence remains. They continue to divide, though more slowly. They continue to repair, though less completely. They sustain life for as long as they can, carrying resilience forward even as time erodes strength. Their quiet work ensures that life endures not only in youth but into the long arc of years. They are with us always, from the first moment to the last, unseen but indispensable. It is soothing to imagine stem cells as the foundation stones of a vast structure. Each stone is small, quiet, and often unnoticed, but together they hold the weight of the whole. Without them, the structure would collapse. With them, it stands firm, able to withstand the stresses of time and circumstance. Stem cells are those stones, placed throughout the body, sustaining its form, repairing its cracks, and allowing it to rise and endure. As the reflections on stem cells draw to a close, what remains most striking is their quietness. They do not seek attention, nor do they reveal themselves easily. Yet they are the reason life begins, the reason it continues, and the reason it heals. They are the foundation not only of biology, but of resilience itself. To know them is to know something profound about life. That renewal is always present, that potential is never entirely lost, and that continuity is built not through stasis but through change. In the end, stem cells remind us of a calm and comforting truth. Life is not fragile because it changes. It is strong because it renews. Stem cells are the silent evidence of that strength, the quiet foundation upon which every heartbeat, every breath, and every thought rests. They are not loud, not dramatic, not insistent. They are steady, persistent, and enduring. To reflect on them is to rest in the assurance that renewal is written into life itself, always present, always available, always at work, in silence, from beginning to end.